Hi everyone, a very good evening to all of you. Welcome back to the Nishchay series lecture, and I am back with the lecture 2, the second lecture for all of you, those who have joined. Please drop a hi, hello, good evening in the chat box so that I know that you have joined us. Eagerly waiting for all of you. It was amazing interacting with all of you in yesterday's session. Okay, so a lot of you have joined on the app. So first time meeting all of you. So hi Smruti Ranjan, hi Sophia, hello, hello Manish, hi Rishabh, hi Chiranjeev, good evening. Can I have the app also? Yeah. Good evening Bunny Brata, good evening Raman, good evening Risha, Krishna, and of course, okay. Great. Awesome to see all of you, and I hope a lot more of you are going to be joining us today. So today we start with the actual real. Okay. Ma'am, should we make notes or add points to printed notes? Good evening. Good evening. Good evening. So this lecture is involves a lot of pictures and involves a lot of videos and everything. So for your imagination, for your 3D imagination. And also this lecture also has a story, because as promised in the yesterday, I'm going to tell you one little story which is going to encompass all the important scientists of microbiology, so that you don't have to learn each one of them. So I would suggest that whichever little point you feel that you should note down, because it is fact heavy, you can make small notes. If you have existing PW notes, then just simply annotate on top of them. It should be enough, right? Okay, good evening. A lot of you, good evening. Great, amazing. So today we are going to start with the general microbiology, and today is actually the first ever lecture, and we are going to start with the scientists and microscopy. The microscopy is what we are going to start with. Now, yesterday I had asked a question. The food for thought was that what is a doctor? And I was very happy to see that a lot of you did respond. You got onto my social media and you commented, and I got some great responses. Now the question was asked in response to one of the students asking that should review books be read right from the beginning, or standard books should be read. So basically a lot of you said that a doctor is somebody who takes care of the mental as well as the physical health of the patient. A doctor is one who's full of empathy. A doctor does not just have clinical skills, this and that, etc. Amazing. It was really amazing to see that your generation is really understanding the essence of being a medical doctor or a medical practitioner. But the reason I asked this question was to be able to differentiate between the two things. So there is something called as a healthcare provider and a medical practitioner, and then there is somebody called as a doctor. A lot of the times the doctors are confused or interchangeably thought of as healthcare providers. But the fine difference is that a healthcare provider is everybody who works in the healthcare system and who gets in contact with the patient and who provides that care and support and everything, and also helps in diagnosis of the patient and everything. So all of the staff that you see in the hospitals. But a doctor is also a subject matter expert. So you also have doctors of physiology, history, and humanitarian sciences and social sciences. Who are they? They are the subject matter experts. So you are also getting the title of a doctor, so you're getting the dual responsibility of becoming a healthcare provider as well as a subject matter expert. Your opinion, your clinical acumen, your decisions are final for every patient. So if you want to go ahead and make those final decisions for your patients based on some review books or guides, then you're most welcome. Otherwise, the internationally accepted standard books or even Indian author books, which are directly derived from these international standard books, is what I expect that you are going to read. And also taking all the help that you need. That is why we are here, to help you, to make you understand the concepts, give you the right knowledge so that you're not distracted, you're not misinformed, you're not ill-informed, and you become the subject matter experts in the true sense of the word, right? On that note, and continuing from the last class, hi everyone, a very good evening to all of you. I might not be able to say hi to each one of you individually, but I can see a lot of you have joined, and it's amazing to see all of you today. Right, so we jump into microbiology, and this is going to be our approach for the topic subject. We are going to start with the general microbiology aspects, and I told in the last class also that why is general microbiology important, because you get an orientation into the world of microbes, which is microscopic inside a microscope, and they have an entire life going on there, and then how we bring their microscopic world into our laboratories, and how we keep interacting with them, and all of this interchange and exchange that happens. It is really necessary to understand their mechanics and behavior in order to understand how we are going to make a diagnosis and how it's going to impact the lives of our patients, right? Immunology is the second aspect which we are going to cover. It is the way our body interacts with these microbes, the defense mechanisms that our body has in fighting these microorganisms. So this is immunology, and that is what we are going to deal with, and this is very, very high scoring, and probably this is the only time that you're going to read immunology at any point, and it will really help you understand how vaccines work, how our body behaves, and even how so many diagnostic tests, what are the principles behind them, how are we utilizing this interaction and in for our benefit. Then system, we'll jump into the systems, the organ systems, the central nervous system, the respiratory system, the gastrointestinal system, the genitourinary system, the skin, etc. Each and every system, organ system of the body we are going to take up, and then we will look at what are these different bacteria, viruses, fungi and parasites which can affect these systems of the body. Suppose you have a patient who comes to you with jaundice, and the liver enzymes of that patient is completely deranged. So what all infectious agents can you think of in such a patient, and then order the right tests for those patients, right? And then of course certain miscellaneous topics in the end, where we are going to take up this entire plethora of knowledge that we have accumulated, and we are going to put it in a systematic manner in the form of tables and some final topics in which we are going to accommodate our entire knowledge of microbiology, right? A lot of you are continuously joining. It is amazing to see that. So welcome all of you. Big welcome. So last time we had left, we had started the history of microbiology and with one scientist, A.V. Leeuwenhoek. In today's lecture, two major topics I am going to cover: microscopy and the certain scientists. This is what we had talked about yesterday, that this really old guy, we do not even have his photo. Every time in exam, you're only going to get a painting. So the painting, where to message for doubts. Okay, so I had shared my social media in the last class. Currently the class is going on, so you can very well write in the chat box if you have any doubts which might erupt during the course of the lecture. Otherwise, after the lecture, you can always reach out to me on my social media. At the end of the class today, again I will share the links with you. Great. So who is he? He is Antony van Leeuwenhoek, and he gave the concept of little animalcules because he was a tech savvy tailor who loved to grind and increase the magnification of microscopes. So he is the father of microbiology, because the first ever person to tell that there is a world full of living things which we cannot see with our eyes, and they're breathing, walking and interacting just like we are interacting. So the first ever in the world, this thing was told by somebody, and he also gave us the idea of creating the modern day microscopes, because he worked with a device which was able to magnify small things into bigger things, right? So father of microbiology, A.V. Leeuwenhoek. Then I also told you that when he told this, nobody in the world cared, and 2 centuries passed just like that. For 200 years, nobody even cared about his contributions. Then I told you after 200 years came these two guys who hated each other, who had the biggest rivalry ever, and it was only because of their rivalry that we could have the modern day microbiology that we know of, and you are getting tortured by this subject in second year of MBBS. Now, this slide looks quite negative, and it's full of hate and everything. So I think let's continue with that note. And since this is the beginning of the lecture and you're quite fresh, and history tends to put people to sleep, and since I am going to tell you a nice little story, I don't want you to sleep off. So what am I going to do is I'm going to leave you hanging. So we will stop the history right here, and in the end of the lecture, when you're tired with a lot of logic I'm going to take up the history and we'll listen to a nice little story while relaxing completely. So I am going to, yeah, okay, Chiranjeevi, you are right partly. He's not Edward Koch, but we'll just see who he is, okay? So, right, so we are going to start with the other topic, which is microscopy, right? So when I say microscopy, you obviously understand that a microscope is a scope means an object we look through or a lens which we help to see objects which are in micrometer or micrometer sizes, okay, which are microbes or microorganisms. Now the different types of microscopes are asked to you. You are supposed to know, understand the principles behind these microscopes, and there are certain googly questions which they throw your way, and then I don't want you to get befuddled by these questions. So very, very logically and as promised, very, very mathematically and using a lot of concepts of physics, we are going to understand the functionings and principles of these different types of microscopes and how they function. And like I had said, that in general microbiology, if you want to read general microbiology, in the end, it gets quite irritating, right? So we will finish it off right now so that you don't have to go back to it. Okay, for a lot of you who are writing that it's showing some error on the app, I would advise that you please log out and log in again, try once again. I hope the team is taking care of it, and if the error persists, you may join on YouTube right now, okay? If it persists, because we have to continue with the lecture because we have two important topics to take care of today, right? All right, so continuing with the microscopes, the first and the very basic one is called as a light microscope, right? A light microscope is also called as a bright field microscopy. What is the meaning of a bright field? You see this image right here. You can see some fungus-like structures, okay? They might look like plants to you right now, but by the end of microbiology, you will realize that these are fungi, fungus, okay? You can see the bright blue color against a white background, a background which is lighter. So that is why it is brightly lit, it is white and bright, and it is the background. This background is called as a field. What is the meaning of a field? When you're looking under a microscope, you see this is a binocular microscope, like we have the binoculars, means two ocular, two eyes you're using to see at something. That is a binocular microscope. In your laboratories, in your colleges, you might have uniocular microscopes, wherein you just have one little round thing through which you peep inside and look at the slide, okay? So you see how this child is looking inside. He's focusing on one particular area on the slide. I'm sure you know what a slide is. Of course, first year MBBS you're done, you know what a slide is, right? The point at which he's looking is going to appear like a circular field to him. So the area in which he's looking is the field of view. When this field of view is brightly lit and is white, we call it a light microscopy or bright field microscopy, okay? Seems quite obvious to you, but this will make sense when I talk about the variations and the different types of microscopes. Like right now, I am standing in a room, okay? This room has a lot many things, but you can't see them because the field of vision of the camera is only till me and this board behind me. So this me and the board behind me is your field of vision. Rest anything else in the room, you're not able to see. Does not mean the other things are not existing. Of course, the other things are also existing. So please understand what a field means. So when that field is bright and white, we call it bright field microscopy. So what is the definition? When you see colored objects, and if this was black, these fungi was black, you could have been able to see black. Colored or dark objects, when you see against a light or a white background, that is light microscopy. This is what a microscope, okay. I will just come on to that. This is what a light microscope looks like. I am sure you have seen this simple bright field microscope in your labs, in your colleges. You might be aware of the certain parts of it also. We are going to cover the parts because they make very, very important viva questions for you, okay? And the way these questions are asked, you might seem like, oh, I know everything. I know this is stage. I know this is this and that. But the questions that they ask you sometimes feel like googlies, okay? What is this examiner asking me? So taking all of those questions in mind, We are just going to understand the mechanics and the principles and the functioning of this microscope, so that A, you can ace those vivas, and B, when you are focusing the slides in the time of exam in your practical halls, you forget to move the condenser, to change the objective, to actually control the light which is passing through the lens, and then you're panicking and you're like, ma'am, focus Neura, what do I do? So to prevent all of that, we have to understand the logics, the physics and the mechanics of these microscopes. You understand a microscope is an instrument which I am using to magnify these small little microorganisms so that they appear big enough, not so big, but just big enough so that my eye, human eye, can now see them. Right, that is the function. So you must have, with your existing knowledge, you must understand what is a resolving power. So what is a resolving power? So any optical instrument, your eye is also an optical instrument, right? The microscope is also an optical instrument. Anything which is an optical instrument which basically transmits the rays of light and we see things has a resolving power. What is a resolving power? The ability to see two objects as separate, right? This is my two fingers. They are close by, but you can still appreciate the small little gap between them so that you can call them as two separate objects. That is the resolving power. I hope you can all appreciate this little dot on the blackboard, right? This is one single dot. Do you all agree? If I can keep having some comments, and you can keep writing in the chat box, then it would be nice. Then I know that you're interacting, right? If you see these little dot, okay, great, good to know. What, now this is a single dot, right? What if I zoom in? What if I zoom in? If I zoom in, you can see that this is in fact many small little dots. Okay, this wasn't a single dot. So what did we do? Just like with a microscope, when on a slide we cannot see anything, and when we zoom in, we saw that it's a collection of dots. Initially we could see that it was a single dot. So now at that far a distance, or when the size of this dot was so small, your eye could only tell you that it's a single dot. But when I zoomed in, you can appreciate that these are in fact multiple small little dots. So this ability to see this dot and this dot as separate is the resolving power. Okay, so when I say that, okay, you can see this dot and this dot as separate, because you can appreciate this small little distance which is there in between these two dots. That is why you are able to see. And what is this distance called as? This distance is called as the limit of resolution. So the power of the optical instrument to tell two closely set objects as two separate objects is the resolving power, whereas this distance between the two objects is called as the limit of resolution. So if this limit is larger, that means the resolving power is lesser, right? Obviously. Now we all understand that there is something called as the wavelength of light. And this resolving power is always inversely proportional to the wavelength of light. That means the lower the wavelength you are using, the better is the resolving power of the microscope. So when I am using a bright field or a light microscope, what am I using? Or the microscopes that you know of normally, these bright light microscopes, they are using a white light, right? So this is the visible spectrum of light we are talking about, 380 nanometers to 700 nanometers, right? All your neat UG knowledge only I am speaking about right now. So this bright light, white light is in a certain spectrum, and then the light which is lower in the wavelength spectrum is the UV light. So the microscopes which use UV light have a better resolving power. Okay, so we will come on to that. First, let's focus on a light microscope and the resolving power we were talking about. We have all seen this scale. This is simply an image of a ruler, right? The ruler that you use, the 15 centimeter scale that we use. We all know that each segment is if this and this is 1 centimeter, I know that each small line, the distance between each small line is 1 millimeter, right? So if I put a single little dot in the space between the millimeter, can you appreciate it? You can appreciate it. So your human eye, without any aid, now aid can be a spectacle. that is still a part of you only. An aid means a microscope. Without any aid, if the human eye can appreciate how much, 0.2 mm. 0.2 mm, and this becomes a previous year question. Okay, this becomes an MCQ right now. 0.2 mm is the resolving power or the resolution of human eye, right? What about the microscope? For that, we have to understand the relative sizes of these different microorganisms. So you see, what is this mycelia? I am sure from your NEET UG knowledge, you know what mycelia is. It is that fungus which grows on our breads and which grows on our walls. So we are able to see it with our naked eye, right? So the size of these fungi is quite large. It is in mm. It is around 100 micrometers, which is approximately 1 mm, 0.1 mm. So we are able to appreciate with the naked eye. As we go down the spectrum and we move on to the yeast cells or the bacteria, we come on to 1 to 10 micrometers size. Okay, one bacteria which you are going to see very, very soon in your labs is Staph aureus, Staphylococcus aureus. The size of that bacteria is 1 micrometer. Right, so we have to magnify it 1,000 times to be able to see it. Okay. So whenever we are using a light microscope, around 0.2 micrometers, the smallest bacteria is called as a mycoplasma. We will read about it, don't worry. So the smallest bacteria which can be seen with the light microscope, the resolving power is around 0.2 micrometers. So we have just seen unaided eye, it is 0.2 mm. When I talk about light microscope, again a question, it becomes 0.2 micrometers, right? 0.2, 0.2, but now it has become micrometers. And viruses, which are even smaller, okay, even light microscope is not able to make us see them, magnify them enough. So for that we need an electron microscope. Now we have gone into 0.05 micrometers. So now in nanometers we are talking about, okay. So most viruses are around 100 to 300 nanometers in size. That is the size of a virus. So electron microscope, which uses electrons instead of light, the resolving power becomes 0.2 to 0.5 nanometers. Nanometers, another question, done and dusted. So we have covered a few MCQs also for you, right, with this resolving power. Now, we are going to look at all the different variations of a light microscope, and we are going to look at the electron microscope as well. So don't worry. Let's come back and look at this microscope. I am sure all of you are aware that this is the body of the microscope. This right here is the source of light. This is the source of light. What is the source of light? It's a simple bulb. These are LED bulbs which are used, just like the bulbs or the lights that you must be sitting under in your rooms right now, just smaller. Okay, they are very, very small, of a lower power. White light they emit, just like all the lights in our room, they all emit white light. The white light goes to this stage. This stage is where I put my slide, my object, the slide with my bacteria, the slide with my specimen, the slide with my object, right? These are a group of lenses, and since these lenses are focusing on the object on my slide, these are called as objective lenses. Objective lenses. Then there is a tube through which, so light is coming from here, entering the objective lenses, passing through this tube, and here is where you look. So when you are looking, here is your eye. So what is this now? This portion is called as the eyepiece. Eyepiece. This eyepiece has a lens. The eyepiece has a lens which has a fixed power, and magnification is 10x. 10x means 10 times. This eyepiece magnifies anything 10 times. So if I put an object over here, and the light which is entering through the eyepiece, the object will be magnified 10 times. And this is fixed for all microscopes, and no matter which objective lens you are using, this 10 times magnification is fixed. Now what is this little dial over here? Let's zoom in. So this is nothing but my light source. Now what have I done? I have zoomed onto this part of my microscope, and I can see that the light is going to come and fall like this onto my stage from this bulb. And what is this over here? This is a dial, the dial which I showed you. It has two things. There is a bigger dial which I can move like this, and there is a smaller dial which I can move like this. Okay. So this bigger one is called as the coarse focus. Coarse focus. And this small one is called as the fine focus. Fine focus. So what happens when you move the coarse focus? When you move the coarse focus, the stage goes up and down. As the stage goes up and down, the object is either brought close to my objective lens or taken farther away from the objective lens. So at a point that I find that my eye can just about appreciate the image, I will stop. And then I will start using the fine adjustment to further clarify the image. I am sure you have used a simple microscope in first year MBBS also, and you already know what are the things that I am talking about, right? With this orientation, I can now talk about these lenses. Now these group of lenses, this is called as a nose piece because it has all of these different objective lenses. Yes, a lot of you are writing also, so it's very nice to see that you are aware what I'm talking about. These objective lenses have different magnifications. Okay, this is just another image showing you the same thing. So magnification, I told you eyepiece magnification is fixed. It is 10 times. What is the magnification of these lenses? This lens is a 10x lens. What does that mean? That this lens is going to magnify my object 10 times. This over here is a 40x lens. So this lens is going to magnify by 40 times. This at the back is a small one, 4x. And then there is another one which is 100x. So the possible objective lenses are 4x, 10x, 40x and 100x. 100x. Right? These are the powers of the microscope. What is the 40x, 4x called as? It is simply scanning. This is called as a scanning lens. Scanning means simply you are just browsing through and seeing if at all there is something on the slide or not. Then the 10x is a low power. You just want to see certain bigger objects and you only want a little bit magnification. Then this 40x is called as a high power lens. High power because now you are magnifying something higher, and this over here, the highest possible magnification lens with a light microscope is not called high power, it is called as oil immersion, and we will just see in a little bit why it is called as oil immersion and why we use oil. Where did this oil come from? Okay. So what is the total magnification achieved? Please do not forget this 10x of the eyepiece lens. So if you are using a 4x power objective lens, if you're focused, like this person is focused the 4x lens onto the slide, then 10x of the eyepiece also you have to take into account, because when the light is passing onto your eye, it is passing through this 10x of the lens, it is passing through the eyepiece, again it is meeting the eyepiece lens and it is getting magnified 10 times further. So the total magnification achieved when you're using a 10x objective is 10 into 10, 100 times. When you are using a 40x objective, 40 into 10, 400 times. So the final magnification achieved is 400 times magnified. So if you are using the 100x objective lens, then the final magnification achieved is actually 1000 times. So the question that they will ask you, what is the maximum magnification achieved using a light microscope? It is 1000 times magnification. So whenever you focus your gram stain or any of your stainings which we do, you have to say that I can see this and this bacteria. We will discuss about all the details at 1000 times magnification, right? Now this magnification, now why was I calling this the oil immersion? Oil immersion. See, this is suppose this is your 100x objective lens. Okay, this is a slide, this is a cover slip. There is always an air gap between the two, right? So the light which is going to come from the bottom, the light is going to fall over here, fall onto the cover slip, and then you know that this glass, this slide is made up of glass, the lens is made up of glass, they all have their own refractive index. And the refractive index of the air gap in between these two is going to be different from the refractive index of this glass slide and this glass lens. So what will happen after refraction? Your rays of light are going to get refracted away. So all the light is not reaching your objective lens now. Some of it is getting deviated and refracted away because of this air gap. Okay, this thing is happening even when you're using the lower powers of the microscope, but then that is fine. Why is that fine? I will tell you. Basically, just that in the 100x objective lens, you don't want any light to go away. You want all the light to reach you. Why do you want all the light to reach you? Because when you're using a high power of lens, you are usually using it to look at stains. Stain simply means colored dyes which I have poured on top of my bacteria in order to appreciate it better. So let's look at one image. You see this chair? Yes. This is a brightly lit room. This is probably morning time. So you can appreciate all the colors vividly and nicely. You can appreciate the red of the chair. You can appreciate the green of the wall behind the chair. What about this one? This looks like evening time. Now the light is less, okay? The most of the light is not reaching this room. So what are you seeing? You can barely tell the color of the background. You can barely tell if the chair is pink or it is red. So this is called as a contrast. What is a contrast? When you are able to differentiate between the two colors. So when you want to look at colored objects, you want maximum illumination and brightness. Now if you are losing out all the light to refraction, barely anything is going inside. So you want to avoid that. Similarly, here I have shown you this is the source of light and not all the light is reaching the lens. It is getting refracted away. So what can I do? I need to fill this air gap. I need to fill this gap. So I need something whose refractive index is the same as that of the lens and that of the slide. So what can I use? I can use immersion oil. What is this? This is immersion oil. Why am I calling it immersion oil? Because I am going to immerse or dip my lens, my objective lens, I am going to dip inside this drop of oil. So I will put this drop of oil onto my slide, and what will I do? My objective lens, I am going to dip inside it. So you can see now that this is the glass slide, this is the oil, and the lens is immersed or dipped inside it. That is why it's called as oil immersion lens, because lens is immersed inside the oil. Now what is happening? The light is coming and it is passing straight through, because now this entire thing has become like one single medium. Air has been removed, refractive index of slide, refractive index of oil, refractive index of lens, everything is same. So this is now one complete medium, and now no light is getting refracted. So all the light is passing straight into my objective lens and straight to my eye without losing any light. Right? So you have oil immersion lens. What next? I was talking about contrast with you. You can appreciate this image or not. So magnification you have understood. If you get any questions in exam, what is magnification, what is the final magnification, these are all PYQs. You can definitely attempt these questions very, very nicely. Now contrast I was talking about. Can you focus on these two images here? So what is this? This is a white box against a white background. And over here, you see there is a lot of light. High brightness is there. So you can barely appreciate and see this box kept here. If you just look at it in one go, you will miss the box, okay? What is happening here? Here the light has been dimmed a little. Now there is a contrast between the background and this box. Now even though the background is also white, the box is also white, you can still appreciate the outline of the box because the light has been dimmed, because the contrast has been achieved by playing with the light that I am allowing to enter. Right? Similarly, with this colored image, you could see that you need light to appreciate the colors. When the light is dim, you cannot appreciate the colors. Over here, when you're looking at transparent objects or white objects against a white background, you need to dim the lights. Okay, so that is now the role of a condenser. What is a condenser and a diaphragm? You see, this is again the bottom of the microscope. You can always find the stage. Always, whenever you want to know where I'm looking, look for the stage. Now, this is the place beneath the stage. This right here is my light source. Okay, this is emitting light. And what is she holding? This is like a aperture or a window. Okay, a khidki. And this entire thing setup is called as a condenser. We looked at it in this image also. This one right here at the bottom of the stage is called as a condenser lens. What will a condenser lens do? As the name itself says, it is going to condense all of the light and throw it onto the object. So you know that the source of light is beneath the condenser, And where is the object kept? Above the condenser. So it is going to gather all the light and throw it all onto the object. That is the function of a condenser. Now, why have I placed this window on this condenser? This window, so this is the condenser lens. It is going to take up all the light and condense it from here onto my object. And inside this condenser is this aperture diaphragm. Aperture, diaphragm, iris, anything. Okay. This is like a window. So she was moving this little lever, and as she was moving this, like the shutter of a camera, this was opening and closing. So what will happen when this will open and close? I can show you. As just like the shutter of a camera opens and closes, your diaphragm in your lens also opens, and when it opens, lots of light goes onto your objective lens, and when it closes or it is just about open, only a little bit amount of light is going to enter your objective lens. So what do I want? When I am trying to look at such an image, I want my aperture to be fully open. Like it was in the beginning. And when I want to look at such an image, I want less light, so I want the aperture to be just open, which is somewhere like this. Just a little bit is open, only a little bit amount of light is opening. So now then they will ask the question, that is the mistake that you make in your exams, and then you make blunders and get scared. When are we going to have such a situation in the lab where we want to look at white objects against a white background? When we want to look at transparent bacteria. So I will show you this image. Here you can see certain transparent bacteria against a transparent background. Over here you can see the bright red bacteria, and there is no problem in identifying these. But when you want to look at such an image, it becomes a problem. So then you want to have a low power of the microscope, and whenever you are using the low power objective lens, you want minimum amount of light to enter. Ma'am, why don't we put a stain on these bacteria? Because whenever we put a dye, it's a chemical. The bacteria dies. So the behavior of the bacteria, whether it is motile means to move around, whether it is moving around or not, or whether it is a bacteria which does not have the ability to move around, whether it is, if it is moving around, how is it moving? Is it moving like a snake, or is it moving like a tumbling barrel, or is it, so all of these motility we are going to discuss as we discuss microbiology, okay, as we get into the main subject. But we want to appreciate the motility of the bacteria, and we cannot pour chemicals on the bacteria to see the motility, because it will die. So at that time we have to see, and by nature bacteria all are colorless only, they don't have their own color. So to be able to see these white or light bacteria against a light background, I need to use the low power of the microscope. So when I am using the low power, this is something how the condenser is going to look like. Whenever I am using a low power lens, such as a 4x or a 10x lens, I want only this much light to pass. So where is my condenser going to be? It is going to be the lowest, and the aperture is going to be just open. Whenever I am using a high power such as 40x, I want the condenser to be somewhere midway and the aperture to be opened midway. When I want to focus at 100x or oil immersion, I want all the light in the world. So I will take my condenser topmost, I will open the aperture all the way, and I will let all the light to pass to my objective lens. So in exam, if you are focusing a hanging drop or motility slide, make sure that you are focusing on a 10x or a 40x, and your condenser or aperture are accordingly placed. And whenever you are focusing a gram stain slide, make sure your condenser is uppermost and aperture is fully opened, because then you end up saying, ma'am, I can't see anything, my entire slide is dark. I performed the staining well, but the entire slide is dark, I can't see anything. Why? Because you have shut down the light. Okay, so please ensure all of this. At this point of time when you're relaxed, all of this seems like obvious nonsense that I'm talking about, but these are genuine mistakes that you make. So if you will understand the concepts, you will not, this is not something that you will have to remember now when you're performing the practical. This is something very logical, and it should make sense to you, right? Now, we have discussed bright field microscopy in detail. Now there is something called as dark field or dark ground microscopy. We understand field or ground can be two words which can be interchangeably used, because ground simply means background and field simply means the field that you're looking at. Now why do I have this picture? So this is a person on a stage, and just like we have a spotlight on the stage, what does the spotlight do? Currently, the room that I am standing in, there are lights on me so that you can see me and you cannot see the other things in the room. Similarly, on the stage, whenever you have an actor performing, there is a spotlight on top of her, and if you look closely, there is actually a person standing here. I hope you can appreciate. If you can appreciate, please drop a yes. There is actually a person standing here. Okay. Which bacteria are shown in the picture, you will know very, very soon. You will know what bacteria are shown. But since the spotlight is not on him, you cannot see him. You can only see her. Similarly, you can see over here, this dancer has a spotlight on top of him. So this is playing with the light. What did we do? We created the room completely dark, and the only thing that we wanted to look at, we have put light only on that thing. Now, what is this type of a light which is reaching you? This is a reflected light, of course, right? Because the light is only falling on her. It is getting reflected and reaching your eye, and only that reflected light is what is reaching your eyes. The other light which is being transmitted from the light source and everything, you're not able to see. So in a dark ground microscope, what is the type of light we use? We use a reflected light if they ever want to ask you this question. In a light microscope, what were we doing? We were simply lighting a bulb and making the entire field bright. Over here, we are only putting the light onto our object. If this girl now turns into a bacteria suddenly, so imagine that the answer is Michael Jackson. No, this is a dancer impersonating Michael Jackson, okay? Probably his dance style, but this is not Michael Jackson. So suppose this girl turns into a bacteria. So now you will be seeing dark colored background and only the bacteria shining. This change that we have now made is dark ground microscopy. Now up till now you have seen many beautiful 3D images and everything. So let me take you into the 2D spectrum. Let's adjust our eyes and brains to be able to understand these ray diagrams. So this is a simple light microscope. What happens? You have the source of light, then you have the condenser lens which condenses all the light, puts it onto the specimen. From the specimen it goes to the objective lens, from the objective lens the ocular or the eyepiece lens which was 10x, and finally an magnified image reaches your eye. This is the simple line diagram of a bright field microscope. If they want to give you an extremely easy question paper, then they might ask the short note on theory, otherwise they will not. So my advice would be, don't waste your time practicing this ray diagram, because if they ask you, it's your Diwali, because you can very easily make this diagram any day, right? So now how do I make sure that not all the light reaches? What will I do in a dark ground microscope? I will simply put an opaque light stop. An opaque light stop, like an opaque disc, I have put over here in the path of the light before it even reached the condenser lens. So what did I do? I created a hollow cone of light. Light moves in a conical form, right? So in a cone, if this is moving, in between this cone, I have put a patch stop. So the light only from the periphery of this patch stop is now going above, as you can appreciate over here. This center white portion does not have any light. So now only the light which will fall onto the specimen will get reflected and enter the objective lens. Rest other light will not enter inside the objective lens. All other light has been stopped. So ultimately what will you see? Only and only the object from which the light is reflecting and reaching, and not the light which is there are surrounding the object. Right? So simply we are playing with the light. Over here you can see the comparison. Here the light is going undisturbed, entire light is entering inside the objective lens. But over here, since you have put an opaque disc, this entire thing has no light in between. This is a hollow cone. So only the light which is getting reflected from the specimen is entering inside the objective and reaching. So the final image which is formed in a bright field is, you can see a brightly lit background and you can see the colored or the colorless or the white bacteria. But in a dark field, you see completely the background is dark and only the outline of the bacteria can be seen and nothing else, because only the outer surface of the bacteria is reflecting the light reaching your eye. So this is simply, dark field is nothing but I have taken a normal light microscope and simply put an opaque disc in between the path before it reaches the condenser. Simple. That is what is a dark ground microscope. Now suppose this is a thread, a white little thread. This white little thread is now against a white background and you can barely see it. But if I put it against a dark background, you can very nicely see this little thread. So now just imagine these really tiny and thin threads of microorganisms. What are they called as? They are called as the spirochetes. Spirochetes. Name itself says, don't they look spiral to you? These are spirochetes. Chete means hair and spiro means spiral. These are really thin spiral bacteria, which are so thin that if I look at them under a light microscope, I will definitely miss them because they get lost. And also they have a very special type of a motility, corkscrew motility. We will read about it, don't worry. So this motility I have to appreciate in order to say that, okay, this is a spirochete I am looking at. So I cannot put a stain on them because if I put a stain, they will die. I have to appreciate their motility, and they are so thin, just like this thin little rope, that against a bright field microscope I can barely see them. So the utility of dark ground microscope or dark field microscopy is to detect these spirochetes or these thin tiny spiral organisms. Okay, great, awesome. Now what is the difference between these two images? If I tell you right now, with this knowledge, you will say that this is a bright field microscope image and this is a dark ground microscope image. But I will tell you that both of these are bright field microscope images only. So this is a stained, normal stained slide where you are seeing a red colored bacteria. Over here, what have I done? I have done negative staining. Negative staining. So over here in the previous example, I was playing with the path of light and creating darkness in the field. But over here, I have not played with the path of light. I have simply stained the slide black, and on top of it, the colorless bacteria is shining. If you're confused right now, we will talk about stains in general microbiology, in which we will say that there are simple stains which I am putting on top of the bacteria only. And then there is ulta, opposite staining or negative staining, means the background has been stained black and then the bacteria is appearing shiny against a black background. Over here, the microscope in both the things that I am using is a light microscope only. So do not confuse a dark ground microscope with a negative staining. I hope it is clear to you. If it is not clear to you, then you may please write and I can repeat this aspect. If it is clear, then we can definitely move ahead, right? So negative staining is your staining only the background. But a dark ground means the field you have made dark, you have played with the light of the microscope, right? Okay, great, awesome. Then there is something called as a phase contrast microscope. How to differentiate? You don't have to differentiate because in the lab you will know what microscope you are using. Okay? So you don't have to differentiate. They will not ask you. So simply if they want to ask you dark ground microscopy, they will always give you the image of a spirochete, mostly. Okay? And if they want to ask you, they will not give you the two options because nobody can differentiate. I just wanted you to know that two different concepts can look like the same. Okay, crystal clear, ma'am. Okay, great. Yes, in negative staining, the bacteria are unstained. We are staining the background. So currently, if I am standing behind a black background, so no stain needed to be put on me. You can see my skin against the black background. But if it was completely white or probably yellow like my skin color, then you would have to paint me. So if you paint me, that is staining the bacteria. But you leave me as it is and you paint the background, that is negative staining. You don't paint anything. You leave me as it is. You leave the background as it is. Simply turn off the light in the room. That is dark ground microscopy. That is the difference. Okay? Awesome. Great. Now, what is phase contrast microscopy? This is the most controversial topic. A lot of the postgraduates are also not able to understand what is this phase contrast microscopy. And if you go on to read from your books, I don't think that there is any book which explains it in so much detail. So I'm going to take a little bit of your time and I'm going to explain it to you very explicitly and clearly. What are we doing in phase contrast microscopy? Up till now, we have discussed two microscopes, both in which we are using the white light only, right? And in one, we have just put a opaque disc, on one we did not put anything. In phase contrast microscopy, also we are using white light. This is also a modification of a light microscope only. Over here, what we are doing, we are taking into consideration, but aim is same in both dark ground and negative staining. Actually, aim is not the same, Samarth, because these spirochetes are so small that even negative staining also we will not be able to see them. They are so thin. So, what I would suggest is you stay with us when we read about the staining, and as you will understand the how these microbes function, you will understand. So, basically in negative staining, we want to look at the refractile surfaces of the bacteria. Certain bacteria have capsules which are like envelopes that they wear and sit. So, in that case, we want to look at the refractile nature of the capsule. So, instead of putting a stain on the capsule, I am putting a stain in the background. What if I don't have the annular disc? But for dark ground microscopy, I am keeping my bacteria alive, and I want to look at the motility. In negative staining, I cannot look at the motility, okay? Because the whole bacteria, actually the whole bacteria can appear white, but the negative staining that we use it for capsulated bacteria, and the dark stain that we pour is not taken up by the capsule of the bacteria. So it's like a lotus. When there is a lotus in mud, even if you put mud on top of it, the mud will slide off the lotus, but the mud will be there on the background of the lotus. Similarly, a lot of capsulated bacteria, even if you put this dark dye on top of them, the dark dye will slide right off their capsules and stain the background black, and they themselves will appear shiny just like a lotus inside the mud. Okay, I hope you can understand now. That is negative staining. But for dark ground, you cannot put any kind of a dye because you don't want the organism to die away. You want to see the motility. So in that case, you are playing with the light. Okay, great. Awesome, good that I could answer you. In phase contrast, what are we doing? We are trying to, if we have blocked the light, only a little amount of light is getting falling onto the object. Like over here, you can see, if you completely switch off the light on the stage and only allow spotlight to fall on the actor, then the light only reflecting from the actor is what is reaching your eye. The light is not getting transmitted to the entire stage. It is only falling onto the actor, getting reflected and entering your eye. So what is your eye seeing? Your eye is seeing, going back, tracing the path of light and falling onto the actor only. If this actor is a bacteria under the microscope, then everything else on the stage is black. Only the light reflecting from the bacteria is entering your eye. So what did you do? You created a hollow cone, just like you created the spotlight, a hollow cone. That hollow cone is created when you put this patch top. So now the path of light is such, initially this complete cone was going, but now only this small little portion is which is only falling onto your specimen and getting reflected. Rest all other light which was simply illuminating the objective lens has been eliminated. Okay, so if I am in a brightly lit room now, so you can see me also, you can see my background also. If we turn off the light of the room and only allow light to fall only on top of me, then you can only see me and not see the background. Got it? Amazing. Great. Please use a little Hindi. Okay, we will see. Right. Okay, I can try. What do we do in phase contrast microscopy? We now want to imagine, we know that light is particle form while light is also wave form. In this, we are using the differences in the amplitude of light and wavelength of light in order to achieve different brightness or intensity of light. If I tell you that this is this wavelength, that wavelength, you cannot appreciate. But you can definitely appreciate between bright light and dim light. So you can differentiate between intensities, right? So if this is a white color and this is a grey color, they are similar stains, but you can differentiate. Why? because the intensity of light is different. The intensity of light emitting from this white and this gray is different, so you can differentiate. That property is what we utilize in a phase contrast microscopy, because we are trying to appreciate the different organelles inside by making the light in different phases. What did I just say? I will just tell you, let me repeat. So this is a cell, okay? Imagine this is a cell. A cell has a dense nucleus inside. It will have some ribosomes. It will have certain internal organelles, etc. Right? All of this is going to be there inside a cell. A light, if let's say a light is in a wave form, a wave of light comes. Suppose it passes through this middle portion where there was no organelle. The light is going to go undeviated through this space. Okay, there will be no deviation in the light because there was nothing stopping in the path of light. है ना? Put bacteria on that margins on that side. No beta, it is not. We are not putting bacteria on the margins. आप रिवाइंड करके यू कैन लिसन अगेन टू व्हाट आई जस्ट टोल्ड यू. अभी हम फेज कॉन्ट्रास्ट पे आ जाते हैं, अदरवाइज वील नेवर फिनिश दिस क्लास. इफ यू स्टिल डोंट गेट इट अदिति, यू कैन प्लीज आस्क द क्वेश्चन ऑन माई सोशल मीडिया एंड आई विल टेक ऑल द टाइम टू एक्सप्लेन इट टू यू पर्सनली. Okay, phase contrast microscope. So the wave of light is going undeviated because nothing is blocking its path of light. Now suppose this was going and it struck the nucleus in its path. Now the nucleus is denser, the optical density is different, है ना? It is more optical dense as compared to these empty spaces. So the light which will come out of this is going to have a different amplitude and a different wavelength. Okay, this different wavelength and different amplitude is what can be perceived as a change in brightness. So simply put, this is what a phase contrast microscope is doing. You're simply putting white light onto it, and depending upon the density of structures inside the cell, it is changing or shifting the wavelength and amplitude, which is appreciated by my eye as a shift in intensity. For the Hindi speakers, जब वेव ऑफ लाइट पास कर रही है सेल के थ्रू, एक ही हमने व्हाइट लाइट ही भेजी है, जब वो ऐसे रास्ते से जा रही है जहां उसके रास्ते में कोई चीज़ नहीं है, वो अनडिविएटेड जा रही है, सेम वेवलेंथ, सेम एम्पलीट्यूड, मतलब कि सेम ब्राइटनेस. लेकिन जब वो टकराती है किसी डेंसर ऑब्जेक्ट से अपने रास्ते में, तो उसका एम्पलीट्यूड एंड वेवलेंथ चेंज होता है और उसकी ब्राइटनेस चेंज हो जाती है. तो एक ही सेल में से एक ही तरह की लाइट डिफरेंट एम्पलीट्यूड और वेवलेंथ में जब बाहर निकल रही है, तो हमारी आईज उसको डिफरेंट इंटेंसिटीज़ के साथ अप्रिशिएट कर रही है. तो मुझे कोई स्टेन नहीं यूज़ करना पड़ रहा, मुझे कुछ इसमें नहीं करना पड़ रहा. अगेन आई एम जस्ट प्लेइंग विद द लाइट टू इंक्रीज द कॉन्ट्रास्ट. कॉन्ट्रास्ट का मतलब हमने अभी समझ लिया, व्हाइट बॉक्स अगेंस्ट व्हाइट बैकग्राउंड, बकवास कॉन्ट्रास्ट. रेड चेयर अगेंस्ट ब्राइट बैकग्राउंड, बढ़िया कॉन्ट्रास्ट. राइट, ओके. करते क्या है, इससे क्वेश्चंस क्या पूछे जाते हैं? टू थिंग्स आर पुट, अ फेज रिंग और अ फेज प्लेट एंड एन एनुलर रिंग ऑन द कंडेंसर और एन एनुलर कंडेंसर. दीज आर योर टू क्वेश्चंस और पीवाईक्यूज फ्रॉम दिस टॉपिक. क्या मतलब हुआ इन दोनों का, इनको हमने क्यों लगाया? जब हमने कंडेंसर के पास, देखो ये लाइट आ रही है सोर्स से, यहाँ पे हमने एक ऐसी रिंग लगाई, एनुलर रिंग, जिसमें बीच में स्लिट है, तो वो क्या कर रही है, एक हॉलो कोन ऑफ लाइट फिर से बना रही है और स्पेसिमेन पे फॉल कर रहा है. तो सिर्फ और सिर्फ लाइट अब मेरे स्पेसिमेन पे ही फॉल कर रही है, जिस तरह डार्क ग्राउंड माइक्रोस्कोप में कर रही थी. अब जब वो स्पेसिमेन पे फॉल करी तो ये वाला काम हुआ, कुछ लाइट अनडिविएटेड गई और कुछ लाइट स्लो डाउन हो गई या रिटार्ड हो गई. ठीक है, तो अब ये रेड वाली है तुम्हारी रिटार्डेड रेज, रिटार्डेड लाइट है, और ये है तुम्हारी अनडिविएटेड, येलो वाली है अनडिविएटेड लाइट. ठीक है, अभी ही आप देख सकते हो कि दोनों का फेज बदल गया. फेज बदल गया मतलब एम्पलीट्यूड बदल गया, मतलब कि ब्राइटनेस बदल गई है. ठीक है, ये वाली इज कितना रिटार्ड हो जाती है जब ये पास होती है, अराउंड λ/4. लैम्डा को अगर हम वेवलेंथ है तो λ/4 से ये रिटार्ड हो जाती है जब ये किसी डेंसर स्पेस से गुजरती है. अब यहाँ से गुजरेगी, स्पेसिमेन से रिफ्लेक्ट होगी, और आईज में जाने से पहले पहले इसके बीच में हमने एक फेज रिंग लगा दी. फेज रिंग क्या करती है, इस रिंग से यहाँ से ग्रे एरिया से, डार्क ग्रे एरिया से पास करती है अनडिविएटेड वाली लाइट, ये येलो वाली लाइट, और ये क्या करती है, इस अनडिविएटेड लाइट को और रिटार्ड कर देती है. तो आप देख सकते हो यहाँ से आ रही है लाइट, येलो लाइट, स्पेसिमेन पे फॉल करने के बाद रेड रेज आर योर रिटार्डेड रेज एंड येलो रेज आर अनडिविएटेड रेज। जब ये दोनों रेज इस फेज प्लेट से पास कर रही है तो जो फेज रिंग से पास करने वाली अनडिविएटेड लाइट है ये फर्दर एक्सेलरेट हो जाती है बाय λ/4, एक्सेलरेट हो जाती है। तो रिटार्डेड वाली λ/4 से कम हो गई, अनडिविएटेड वाली λ/4 से इंक्रीज हो गई। तो फाइनली दोनों का क्या डिफरेंस आया फेज में, λ/2, यानी कि दोनों लाइट λ/2 आउट ऑफ फेज है, दोनों का फेज λ/2 से बदल गया है। इससे क्या होगा, फाइनली जो इंटेंसिटी आई लाइट की, डेंसर एरियाज की कुछ आई और लाइटर एरियाज की कंप्लीटली ऑपोजिट इंटेंसिटी आई। तो फाइनली इमेज कैसे दिखेगी, ये आप देख सकते हो, एक ही सेल के अंदर जो डेंसर एरियाज थे वो डार्कर दिख रहे हैं और जो आसपास के लाइटर एरियाज थे वो लाइटर दिख रहे हैं, या अलग इंटेंसिटी के यू कैन अप्रिशिएट। सो सिंपली यू हैव यूटिलाइज द प्रॉपर्टी ऑफ लाइट मूविंग एज अ फेज, then you have put two important things, a annular ring and a phase ring. Annular ring, the retarded rays are retarded and the undeviated rays are further accelerated by the phase ring, so that both of them are in opposite phases, and when they are in opposite phases then you will see the complete change in intensity and you have achieved an excellent contrast by simply playing with the properties of light without having to use anything else. Okay, if your physics is a little weak like mine, then simply remember that you use an annular ring and you use a phase ring, and ultimately you achieve different intensities of light of different brightness in a single cell. Then you can appreciate the internal structures better. Okay, now we come on to fluorescent microscope. Up till now, the three microscopes that we talked about, we have simply used white light. We have simply playing with the intensity here and there. I told you in the beginning that wavelength and resolving power are inversely proportional. That means if I want to increase the resolving power even more, then I can use a lesser wavelength of light. You see, this is the Vibgyor spectrum, and you know that the visible light is 380 to 780 nanometers. That is the visible spectrum, right? The most convenient microscope to use is a light microscope, because all of these microscopes are a little expensive, but yes, it depends upon the organism. Sorry. So Anish, it depends upon the microorganism what we are trying to see. Some of them are easily visible with a light microscope, so we need not go through all this jazz. But like I said, the spirochetes, if you want to look at the motility, you need a dark ground microscope. And sometimes when you want to appreciate the flagella of the bacteria and the internal structures, like over here you can see these small little spikes which are coming out. This is a amoeba. This is called as a brain eating amoeba, and we will talk about it when we do the general microbiology. For that you might need a phase contrast microscope. So all microscopes have different utilities. Cytoplasm organelle partially black, making them visible. Yes, you're correct. Okay, fine. Fluorescent microscope पे we will come back, वापस आते हैं. अब fluorescent में हम क्या कर रहे हैं, ये जो lesser wavelength UV light है ना, shorter wavelength, इस property को utilize कर रहे हैं to get a better enhanced image. This image is what is a previous year question. अब आप ऐसे कोई फैंसी स्केचेस तो बना नहीं पाओगे, अगर शॉर्ट नोट आएगा फ्लोरेसेंट माइक्रोस्कोप पे जो कि पोटेंशियल क्वेश्चन है, आ सकता है. सो दिस इज द लाइन डायग्राम दैट यू विल ड्रॉ, एंड ऑल्सो क्वेश्चंस PYQ है. ये कॉम्पिटिटिव एग्जाम्स में कभी कभी ये इमेज देके ना लेबल करने को बोल देते हैं आपसे, राइट? लेबल करने को बोल देते हैं, तब आपको पता होने चाहिए पार्ट्स. कुछ नहीं किया, यहाँ से शुरू करते हैं. लाइट सोर्स आई, सिंपल वाइट लाइट ही ली है, यहाँ पे भी कोई ऊट पटांग लाइट नहीं ली है. क्या लगाया, एक एक्साइटेशन फिल्टर लगाया. एक्साइटेशन फिल्टर क्या करेगा, ये आपका विबग्योर आ रहा है, इसमें से इसने यूवी स्पेक्ट्रम की लाइट को कर लिया सिलेक्ट. ये जो ब्लू कलर की यूवी लाइट आ रही है, अब एक सिर्फ यूवी लाइट आ रही है अंदर माइक्रोस्कोप के. यहाँ पे लगाई मैंने डाइक्रोइक मिरर, एंड दिस डाइक्रोइक मिरर इज योर प्रीवियस ईयर क्वेश्चन. लेबल करके पूछा गया था, ये क्या लगा है, नाम है डाई क्रोइक मीन्स कलर, डाई मीन्स टू, मतलब ऐसी मिरर जो अलग अलग कलर के साथ अलग अलग बिहेव करती है। क्या अलग बिहेव करती है? यूवी लाइट क्या है? शॉर्टर वेवलेंथ की लाइट है ना। जब भी इस पे शॉर्टर वेवलेंथ की लाइट फॉल करती है तो ये उसे रिफ्लेक्ट करती है, इट रिफ्लेक्ट्स इट, सेंड्स इट अवे, and whenever a larger longer wavelength falls on it, it allows it to pass through. अब इस बात को हमने कैसे यूटिलाइज कर लिया? हाउ डू वी यूटिलाइज इट? वी आर अलाउइंग द यूवी लाइट टू फॉल ऑन द मिरर, get reflected, and here I have kept my specimen. This is my slide on which the specimen is kept. Okay, there are certain organisms which throw out this fluorescence by their own, like you have the fireflies ना, वो जो fireflies होती हैं, bioluminescence आपको पता होगा, कुछ insects होते हैं जो अपने आप चमकते हैं, ऐसे कुछ बैक्टीरिया होते हैं, और जो अपने आप नहीं चमकते उनके ऊपर मैं कुछ ऐसा सब्सटेंस लगा देती हूँ कि वो चमकने लगे। आपने वो डांसर्स देखे होंगे ना, नियॉन लाइट्स वाले। अब ह्यूमन के पास थोड़ी प्रॉपर्टी है बायोल्यूमिनेसेंस की, पर अगर मैं ऐसी एक ट्यूबलाइट पहन के खड़ी हो जाऊँ और लाइट बंद कर दूँ तो फिर मेरी पूरी आप आउटलाइन देख सकते हो। इसी तरह के मॉलिक्यूल या डाई को, विच इज़ अ फ्लोरेसेंट डाई, मैं टैग कर दूंगी अपने ऑर्गेनिज्म पे। तो फ्लोरेसेंट डाई को मैंने टैग कर दिया अपने ऑर्गेनिज्म पे। अब उस डाई के ऊपर फॉल कर रही है शॉर्टर वेवलेंथ की यूवी लाइट। डिपेंडिंग अपॉन कौन सी डाई मैंने यूज करी है, एक पर्टिकुलर वेवलेंथ एमिट आउट होगी। कौन सी वेवलेंथ एमिट आउट हुई? लॉन्गर वेवलेंथ। लॉन्गर वेवलेंथ एमिट हुई तो डाइक्रोइक मिरर क्या करेगी? शॉर्ट को रिफ्लेक्ट करती है, लॉन्ग को पास थ्रू करा देगी। लॉन्ग विल पास थ्रू राइट थ्रू द मिरर एंड गो अप। Now while it is going up, it will pass through an emission filter, ताकि अगर कोई और wavelength भी pass हो गई है तो उसको हटा देगा, and finally a single color of a longer wavelength light is going to reach my eyes. Detector is nothing but your eyes, right? So what is happening? You have put UV light, it is falling onto an object stained with a fluorescent dye. Finally a changed wavelength, a longer wavelength is entering your eye. So what do you see? You see these fluorescent bright colored bacteria. So you can see the beautiful bright green, the bright orange colored organisms against the darkness. So this is very, very highly contrast. There is no way you can miss this. Okay, this is very bright, this is beautifully, this can be seen. Dichroic mirror, I just told you, di means two, croic means color. This is a mirror which behaves differently with different colors of light. So if a shorter wavelength falls off it, it reflects it. If a longer wavelength falls on it, it lets it pass. So when I am putting from my light source, UV light is coming, so dichroic mirror is reflecting it, and wherever it will reflect, there I have put my specimen. After hitting the specimen, the longer wavelength which is coming out will pass right through this dichroic mirror and reach my eye. So finally, the wavelength which you put on the specimen was a shorter wavelength UV light, and the light which is entering your eyes is a longer wavelength light, which is the visible spectrum light. So what you put was UV, and what you see is either red, yellow, green, depending upon the dye that you used. So you put a shorter wavelength and you see a longer wavelength. That is entirely the principle of fluorescent microscope. Question they will ask, dichroic mirror. Okay. So different dyes can be used, such as a dye which is called as auramine, rhodamine dye, which is used for the TB bacteria. For the TB bacteria, this is a question that they can ask you. Then acridine orange dye, acridine orange, which is used for the malaria parasite, which is plasmodium. I am not telling it right now because you might not understand, but when you will read further, all of this will get revised. So this is a question, this is a question. And then the dye which is used to see fungus, calco fluor white. Why? Fluor is fluorescent, calco is the chitin layer. कोई बात नहीं, पढ़ेंगे, don't worry right now. These are the three dyes which are used for fluorescent microscopy, and these are your questions. Finished, done, dusted. Finally, after a long lecture, we come to the electron microscope. So up till now, what were we using as a source of illumination? Light. End of story. Now, the viruses, which are so small in nanometer, no matter the wavelength of light I use, no matter what I do, They are so small that the light microscope, the fluorescent microscope, the phase contrast is not able to magnify. Now I need to change my source of illumination. अब light से काम नहीं चलने वाला है, अब लाने पड़ेंगे इलेक्ट्रॉन्स. तो ये ऐसा माइक्रोस्कोप जैसा नहीं दिखने वाला, कुछ बहुत ही सोफिस्टिकेटेड मिसाइल जैसा दिखने वाला इंस्ट्रूमेंट इज योर इलेक्ट्रॉन माइक्रोस्कोप. कभी कभी इमेज देके आपसे डायरेक्ट क्वेश्चन पूछ लेते हैं, व्हाट इज दिस? दिस इज एन इलेक्ट्रॉन माइक्रोस्कोप. अब लाइट डालने की बजाय मैं इलेक्ट्रॉन्स डालूंगी. तो ये क्या है? आपका सोर्स ऑफ इलेक्ट्रॉन. यहां से इलेक्ट्रॉन्स आएंगे नीचे. बीच में एक इवैक्यूएशन चेंबर है, क्योंकि इस माइक्रोस्कोप को चाहिए वैक्यूम. वैक्यूम नहीं क्रिएट करोगे तो एयर में इलेक्ट्रॉन्स डिस्पर्स हो जाएंगे पार्टिकल से टकरा टकरा के. वो सब आपको नहीं कराना है. आपको टोटली पार्टिकल फ्री एनवायरनमेंट चाहिए. आप एक वैक्यूम क्रिएट करोगे जिसमें आपके इलेक्ट्रॉन्स जाएंगे. सैंपल पोर्ट यहाँ पे सैंपल रखा जाएगा, ठीक है. सैंपल पे इलेक्ट्रॉन्स टकराएंगे. फाइनली नीचे आपका डिटेक्टर है. यूजुअली हम ये, आप वैसे तो बाइनोक्यूलर्स भी हैं, बट यूजुअली हम ऐसे कंप्यूटर स्क्रीन पे इसको इमेजेस को देखते हैं. कैसे होगा? दो तरह के इलेक्ट्रॉन माइक्रोस्कोप्स हैं. सेम मतलब स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप. मैं माइक्रोस्कोप ऐसे लिखती हूँ, माइक्रोस्कोप, ठीक है. व्हाट इज टेम? ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप. बुक्स में और बहुत तरह के माइक्रोस्कोप्स दिए होंगे. यूजुअली यही दो पूछे जाते हैं, इसके अलावा कोई पूछेगा नहीं, किसी के पास टाइम नहीं है, ठीक है. स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप क्या कर रहा है? स्कैनिंग करा रहा है. सिर्फ ऊपर ऊपर की सरफेस बताएगा, टोपोग्राफी बताएगा. मतलब कि मैं खड़ी हूं, मुझे ऊपर ऊपर से स्कैन कर लिया और मेरी शेप आपको बता दी. ट्रांसमिशन क्या करेगा? थ्रू एंड थ्रू. मतलब मेरे दिमाग से पियर्स होके गया और मेरे दिमाग के एक एक न्यूरॉन्स को आपको दिखा दिया. दैट इज ट्रांसमिशन विच इज पासिंग थ्रू एंड थ्रू द बैक्टीरिया. अब इलेक्ट्रॉन का सोर्स ऊपर से आएगा, एनोड कैथोड कुछ भी लगाओ, डज नॉट मैटर. क्या इंपॉर्टेंट है यहाँ पे? लेंसेज कौन से? ग्लास लेंसेज थोड़ी इलेक्ट्रॉन्स को आपको रिफ्लेक्ट करेंगे, वो तो लाइट पे काम करते हैं. दीज आर इलेक्ट्रोमैग्नेटिक लेंसेज. ऑल दीज इलेक्ट्रोमैग्नेट्स विच आई पुट, दे आर गोइंग टू फोकस दिस बीम ऑफ इलेक्ट्रॉन्स. इलेक्ट्रोमैग्नेट्स लगा दी हमने लेंसेज की जगह पे, और वो इस बीम ऑफ इलेक्ट्रॉन्स को लेके और डायरेक्ट बॉम्बार्ड करेंगी जो नीचे रखा है हमारा स्पेसिमेन स्टेज के ऊपर, ठीक है. यहाँ पे ग्लास स्लाइड्स यूज नहीं होती हैं, यहाँ पे मेटालिक स्लाइड्स यूज होती हैं. मेटल चाहिए हमें, तभी तो इलेक्ट्रॉन टकराएगा और स्कैटर होगा. और जब टकरा के स्कैटर होगा तो यहाँ पे मेरा डिटेक्टर है जो उसको पकड़ लेगा, ठीक है. जब भी किसी एटम पे इलेक्ट्रॉन लगता है तो वो उस एटम के इलेक्ट्रॉन को नॉकआउट करता है. तो ये जो नॉकड आउट इलेक्ट्रॉन्स हैं ये फॉल करेंगे मेरे डिटेक्टर के ऊपर और उसको मैं डिटेक्ट कर लूंगी. तो क्या हो रहा है? टक 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 इलेक्ट्रॉन्स आर बॉम्बार्डिंग, दे आर गेटिंग knocked out, falling onto my detector, and an image is being created onto my screen. So I can now see the topography or the external surface of my bacteria. What is happening in transmission? I want to allow the electrons to pass through and through my bacteria. How do I do that? I put two lenses and I put my sample in between the two lenses. Now the light will pass straight through. I will make very very thin slices of my sample and I will put it in between two lenses, and the electron will pass through and through right through the specimen. राइट इससे ज़्यादा डिटेल नॉट रिक्वायर्ड, सिंपली द प्रिंसिपल. अगर आप सेन की इमेज देखो या स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप की इमेज देखो तो आप ऐसे 3D स्ट्रक्चर्स देख सकते हो. ये आरबीसीज हैं आपकी, 3D बढ़िया सी दिखाई दे रही हैं, ऑल देयर कॉन्टोर्स, ऑल देयर एंगल्स यू कैन सी. व्हाट इज हैपनिंग हियर? दिस इज अ बैक्टीरिया. दे हैव मेड सम कनेक्टिंग ट्यूब्स विद ईच अदर एंड वेरी नाइसली यू कैन अप्रिशिएट द 3D स्ट्रक्चर ऑफ दिस बैक्टीरिया. सिमिलरली आप वायरसेस को देखने के लिए यूज करते हैं. टेम में क्या हो रहा है? थ्रू एंड थ्रू जा रहे हैं तो आपको एक 2D इमेज दिखाई दे रही है आरबीसी की, 3D इमेज नहीं दिखती है क्योंकि वो अंदर की स्ट्रक्चर्स को दिखाता है, 3D स्ट्रक्चर्स नहीं दिखाता है. सो 2D इमेजेस यू गेट विद टेम एंड 3D इमेजेस यू गेट विद सेम. इसके ऊपर नथिंग एल्स. पूरा माइक्रोस्कोपी. ऑल पोटेंशियल क्वेश्चंस, ऑल पोटेंशियल वाइवा प्रॉब्लम्स वी हैव डिस्कस्ड. इसके ऊपर कुछ नहीं आने वाला, स्पेशली माइक्रोबायोलॉजी में तो नहीं आने वाला, फ्रॉम माइक्रोस्कोपी चैप्टर. राइट, सो यू कैन हैव अ ग्लास ऑफ वाटर, you can freshen up. Now we are going to move on to the next topic, and now that your brain is bombarded with all of this information, let's move ahead and talk about the history. The history that we started off with and did not finish, we are going to take it up again now. Okay, great. So happy that you guys are liking it. Thanks for all the nice comments. चलो हिस्ट्री पे आते हैं और हमने Are bacteria friends of ours? Of course, a lot of them are friends of ours. There are the good gut bacteria and the bad gut bacteria. In the gut, there are so many good bacteria that do not allow bad bacteria to grow. They give direct competition to these bad bacteria. That is how we justify all the good bacteria. No, the longer wavelength which is reaching your eye is of the visible spectrum, beta. The longer wavelength is reaching because it is of the visible spectrum. And so you are able to see. And it is of one particular color and brightly fluorescing. Yes? Yes, yes, yes. Okay. Right. Fine. Chalo. Now, I dropped off the lecture over here. I hate you. Right? So, A.V. Leeuwenhoek is the father of microbiology. He discovered the microscopes, called them the little animalcules. This we have talked about. Right? Now, let's listen to a nice little story. Before we go on to the hatred of these two men, let's look at this guy. Who is he? Since 12th standards in school, you know that he is Edward Jenner. Edward Jenner has been known to be the visionary for vaccines. He was the first ever guy who told that if you give the agent to the person, what was he doing? He was living in a village, there was smallpox was rampant. वो देखता था कि the milkmaids who come in contact with the cows and end up getting cowpox, they never get smallpox. So that gave him the idea that if you expose a person to the agent of infection in limited doses, then you can prevent the disease. So he became the idea behind vaccination. So he gave the term variolation, and this is sometimes asked from you. What is variolation and what is vaccination? Vaccination is when you give a little bit of a substance of the same organism into a person, and a lot of us have those BCG scars which we took the BCG vaccine in the childhood. So the vaccination leaves a little bit of a scar. What do you do? Now, vaccination is a more sophisticated modern concept. What Edward Jenner was doing was variolation. He was simply taking a little bit material from the smallpox lesion and taking it and putting it onto the skin of a healthy person. So, this is what variolation used to look like. एक smallpox के patient का थोड़ा सा lesion उठाया और एक healthy patient के ऊपर चिपका दिया. तो जहां चिपकाया वहां पूरा एरिया बहुत बुरी तरह से खराब हो जाता था. बट दिस वाज एंड देयर वर डीप सीटेड स्कार्स विच फॉर्म. दैट इज कॉल्ड एज वेरियोलेशन. अब इन्होंने क्या किया? स्मॉल पॉक्स का वायरस उठा के लोगों को देना शुरू किया, काउ पॉक्स का वायरस उठा के लोगों को देना शुरू किया और लोगों की इम्यूनिटी बन गई अगेंस्ट स्मॉल पॉक्स वायरस. जो काउ पॉक्स का वायरस था उसका नाम था वैक्सीनिया और जो स्मॉल पॉक्स का वायरस था उसका नाम था वेरियोला. दोनों नाम इन्होंने ही दिए थे. ठीक है. अब इस वैक्सीनिया से ही आया द टर्म वैक्सीनेशन. हम क्यों बोलते हैं वैक्सीन या वैक्सीनेशन? क्योंकि जिस वायरस को ये देते थे स्मॉल पॉक्स को प्रिवेंट करने के लिए, उसका नाम था वैक्सीनिया. ठीक है. अब इस स्टोरी को आप जानते हो पहले से ही, एंड हाँ, इस कंट्रोवर्सी पे आएंगे कि पूर्वा शास्त्री में क्यों लिखा है. अभी बात करते हैं. अभी के लिए आप समझो A.V. Leeuwenhoek इज द फादर ऑफ माइक्रोबायोलॉजी एंड देन हु इज लुई पास्चर. अभी देखते हैं. चलो. सो इस कॉन्सेप्ट को समझ के छोड़ दो कि एडवर्ड जेनर ने वैक्सीनिया वायरस दिया और उसने दुनिया को बताया कि भाई हम ऐसा कुछ कर सकते हैं. एक थॉट दिया सिर्फ वैक्सीन्स का. जो मॉडर्न डे हम वैक्सीन्स जिस तरह समझते हैं और देते हैं, दैट थॉट ही कुड नॉट गिव. बहुत पुरानी बात है. फिर आए ये दोनों नफरती डूड्स. Who is he? This grumpy old man. He is Louis Pasteur. Louis Pasteur. तुम में से बहुत सारे लोग, you were also telling me yesterday that this grumpy old man is Louis Pasteur. What did Louis Pasteur do? Louis Pasteur was a French guy. He was a French man. ठीक है. और ये थे एक केमिस्ट. इससे पहले हम इसकी कॉन्ट्रीब्यूशंस पे जाएं, स्टोरी समझते हैं. फ्रांस में बियर की इंडस्ट्री, वाइन की इंडस्ट्री, चीज़ इंडस्ट्री बहुत flourishing industry. Everybody was, the economy was totally dependent on these industries. Louis Pasteur was a chemist. He was a guy of chemistry and he used to work with chemicals. Now, the world at that time was thinking that all of these wine and cheese is formed, the beer and cheese is formed because some chemical reaction happens. When you put the wheat for a lot of time, when you put the milk, some chemical reaction happens और वो cheese बन जाते हैं या वो beer बन जाते हैं. अब ये cheese, beer सब कुछ खराब भी होती थी. So his friend was a big brewer and a big industrialist, and he had his cheese and beer factories, and the cheese and beer used to go bad and people used to fall sick after eating these, and they were called as the diseases of beer and cheese. Now he told him that do something that these cheese and beer, they stop going bad. Okay. Okay, right. So he said that भाई कुछ ऐसा करो that these beer and cheese, they stop going bad, right? So then he took it upon himself to understand the nature of these microbes. Now, while the world did not care about A.V. Leeuwenhoek's work, he cared. इन्होंने पढ़ा भाई A.V. Leeuwenhoek, और उस टाइम पे क्या था, बहुत सारे लोगों ने पढ़ लिया था. पॉपुलर बिलीफ क्या था कि स्पॉन्टेनियस जनरेशन होती है. अब ये जो दो वर्ड्स यहाँ पे लिखे हैं, थ्योरी ऑफ स्पॉन्टेनियस जनरेशन वर्सेस द जर्म थ्योरी. कुछ स्कूल ऑफ लोग थे जिन्होंने एवी ल्यूवेनहुक का काम पढ़ा था. दे स्टार्टेड सेइंग दैट लाइफ इरप्ट्स फ्रॉम लाइफ. बेसिकली वी हैव नॉट ड्रॉप्ड फ्रॉम हेवन एंड वी हैव नॉट बीन क्रिएटेड फ्रॉम फायर और वाटर एंड ऑल ऑफ दैट. वी हैव बीन क्रिएटेड फ्रॉम जर्म्स. Okay, we were all germs in the beginning. These unicellular little forms of life are called as germs. Now, it was against the popular religious belief, तो लोग इसको मानने को तैयार नहीं थे. सबको था कि नहीं, हम तो heaven से आए हैं and we have come from gods and all of this. Now, he took it upon himself to prove this germ theory. He said, I will prove the germ theory. How did he prove the germ theory? He did the very, very famous swan neck experiment. And some old examiners, they love to ask this question about the swan neck experiment of Louis Pasteur. He had a flask whose neck was like a swan. So basically, his flask used to look like this, like a swan's neck. Okay. So, simpler diagram you can see over here. This is a flask and this is a very, very narrow neck it has, which is quite long. What did he do? He put some broth, some beer and all liquid he put inside, and in this he put some yeast. I am sure you know what is yeast from your knowledge. The yeast we use for baking, called as the baker's yeast, it is also one of a microorganism only. He put some yeast inside it. Then what did he do? He heated both the things. Over here he applied heat, over here he applied heat. Okay. He was the first person to realize that all of this wine and cheese which you are making is actually not a chemical process. It is the growth of these germs which is happening, which is why this cheese and wine is forming. But nobody was ready to believe him, so he had to prove it that there are these germs, which are these lives, which are causing all of these changes. So what did he do? Just like that, he heated both of these flasks. What did he do? One had a swan neck, and of the other flask, he broke off the swan neck. Over here, he put a lid, and on this, it was completely open to air. So one flask has a swan neck, and as the lid is completely closed, has a yeast broth, and he's heating it. The other flask, the lid is open, the swan neck is broken, air is entering from here also, yeast is inside and he's heating again. After heating, he realized that in both the broths, the yeast died. The yeast died in both the broths. So he was the first guy to tell that if you heat something, all life dies. And when life dies, we call it sterilization. So he was the first person to say that heat can lead to sterilization. So he gave us the concept of heat sterilization. Then what did he do? Then he removed the heat. He removed the heat from both and he let them sit for some time. After a while, he noticed that in this one, the yeast remained dead even after a few days. New yeast did not come up. But in this one, new yeast had come. New life had erupted. So he said, if spontaneous generation was true, then life should have erupted in both. But only life erupted in the one from in which outside things could enter, and in the one in which outside things could not enter, because one, it is closed from the top, and this tube is so long that no microbe has the ability to reach up till here. Okay, even if something entered, it would stop right over here, could not enter all the way down. So if spontaneous generation was true, life should have erupted here also. Why did it only erupt in the flask which had access from outside? Because there are these germs present in the environment which entered from outside and then again yeast grow in it. So he was the first person who proved the germ theory, and he told the world that life erupts from life. Okay, so a French guy told this. So first question, he is the one who proved the germ theory. Second question, he conducted the famous swan neck experiment and told that this fermentation, this process of this life growing and changing the nature of milk and wheat and converting it into beer and converting it into cheese is actually the process of fermentation. And he was the first guy to tell that heat sterilization is a method, and if you heat things, then they can be sterilized. So he realized that the cheese and beer is going bad because this life or germs from outside is entering and making them turn bad. So he said, why don't we heat these things? And then of course the very, very famous, from the name Pasteur itself, he gave us the concept of pasteurization. So very, very easily now I can write all the contributions. He was the first guy who told us the concept of fermentation. He was the first guy who proved the germ theory, right? He was the first guy who talked about heat sterilization. He was the first guy who gave us the concept of pasteurization, because the name itself says, and a lot of you were also writing in the chat box, pasteurization. And after this, can you imagine? The beer and the cheese industry was just flourishing. So he became a hero. By this time, he was a hero in France. So they named an Institute Pasteur, for him in Paris. So if they ask you a question, the guy who has an institute against his name in Paris, it is Louis Pasteur, right? Louis Pasteur saved the wine and cheese industry. So he is hero now. Okay, Paris. All of the contributions now, you need not rote learn, make mnemonics, nothing you need to make. Right from pasteurization and this swan neck experiment story, you can very easily remember that fermentation भी उन्होंने बताया, germ theory भी उन्होंने बताया, heat sterilization भी उन्होंने बताया, पास्चराइजेशन भी इन्होंने बताया. इसीलिए आप लोग लिख रहे थे बहुत सारी बुक्स, इनको फादर ऑफ माइक्रोबायोलॉजी बोलते हैं. पर पेरिस और फ्रांस का बंदा तो थोड़ा मॉडर्न है ना. एंड ऑल्सो ऑल ऑफ दीज कॉन्सेप्ट्स आर क्वाइट मॉडर्न. आज तक हम पास्चराइजेशन यूज़ करते हैं, आज तक हम जर्म थ्योरी को नहीं डिस्प्रूव करते हैं, और आज तक जो हम लैब में कल्चर्स करते हैं उसके पीछे इनका कॉन्सेप्ट यूज किया जाता है. सो ही इज द फादर ऑफ मॉडर्न माइक्रोबायोलॉजी. सो इफ दे आस्क यू क्वेश्चन, फादर ऑफ माइक्रोबायोलॉजी एवी ल्यूवेनहुक, फादर ऑफ मॉडर्न माइक्रोबायोलॉजी लुई पास्चर. ठीक है, ऑल द कंट्रोवर्सीज सेटल्ड. अब ये क्या है, किस पे काम कर रहे थे? बियर पे काम कर रहे थे ना, तो उसी में तो उगा रहे थे. यहाँ पे भी देखो कुछ तो लिक्विड ब्रॉथ लेके बैठे थे. सो ही गेव अस द लिक्विड कल्चर मीडिया. ही टोल्ड अस दैट दीज जर्म्स एंड दीज माइक्रो ऑर्गेनिजम्स, दीज लिटिल एनिमल्क्यूल्स, यू कैन ग्रो इनसाइड कल्चर मीडिया और ब्रॉथ which is liquid. So a lot of the people like to remember with a mnemonic, L for Louis Pasteur, L for liquid media, if ever you forget. Okay, so from Pasteur all of these things, and from liquid media. Now, why did I tell you the story of Edward Jenner before this? Because Edward Jenner introduced us to vaccinia, and then this guy, he gave us the concept of vaccines. The modern day vaccines in the form that we know of, he is the one who gave the concept of vaccines to us. So sixth contribution will become vaccines. Now which vaccines? Okay, let's rewind a little bit and let's realize, why was this hatred there? Why did he hate this other guy? He is a French man, he is a German man, at the same time, and they hate each other. Why? Because he did not read all of the A.V. Leeuwenhoek's work. He also read it. And both of them started growing these bacteria in their labs. Now, while Louis Pasteur was distracted and focusing on the wine and cheese industry, this guy was focusing on the core microbiology concepts. So, if we have to give the credit of understanding of microbiology entirely, it goes to this guy. This guy is Robert Koch. We will just come on to him in a little bit. Now, this guy was really growing these organisms in his lab, identifying these organisms. ट्यूबरक्लोसिस, टीबी जैसी बीमारी जो हम सब जानते हैं, इन्होंने बताया था कि ये है वो बैक्टीरिया जो टीबी करता है, और उसको अपनी लैब में ग्रो करके दिखाया था, प्रूव करके दिखाया था. ये इतना सारा काम कर रहे थे, बैक्टीरियाज को ग्रो कर रहे थे, और ये जो है उन बैक्टीरिया को उठा उठा के उनके अगेंस्ट वैक्सीन बना बना के सारी दुनिया में नाम कमा रहे थे. तो इन दोनों की राइवलरी चल रही थी. इन्होंने लिक्विड ब्रॉथ का कॉन्सेप्ट बताया, इन्होंने उसको थोड़ा और एडवांस करके बोला लो मैं सॉलिड मीडिया पे उगा के दिखाता हूँ, उसमें अगार अगार डाल दिया. तो इनकी राइवलरी चले जा रही थी, इस एक दूसरे का काम चुरा रहे थे और उसको बेटर बेटर करके प्रेजेंट कर रहे थे. एंड वी हैव द मॉडर्न डे माइक्रोबायोलॉजी ऑल बिकॉज ऑफ दिस राइवलरी, राइट. चलो, अब वैक्सीन्स की मैंने बात करी. 3 ऐसे ऑर्गेनिजम्स या कुछ ऐसे ऑर्गेनिजम्स जो वो उगा रहे थे, ये वैक्सीन बना रहे थे. कैसे याद रखेंगे? बहुत इंपॉर्टेंट एमसीक्यू आपसे पूछा जाता है. क्या है ये? एक गाड़ी है, एक कार है जिसमें वैक्सीन्स रखी हैं. इट इज अ कार ऑफ वैक्सीन्स. इट इज अ कार ऑफ वैक्सीन्स. कौन सी वैक्सीन दी? कॉलेरा वैक्सीन. पर इंसानों का नहीं, फाउल कॉलेरा. ए फॉर एंथ्रैक्स. एंथ्रैक्स की वैक्सीन दी इन्होंने. आर फॉर रेबीज. रेबीज तो हम सब जानते हैं, डॉग बाइट से होने वाली बीमारी इज रेबीज. रेबीज. सो अब आपके 3 एमसीक्यूज फिनिश्ड. कॉलेरा, एंथ्रैक्स, रेबीज. 3 इंपॉर्टेंट डिजीजेस अगेंस्ट विच, yes, useful rivalry, correct, against which Louis Pasteur gave us the vaccine. So just remember, a car full of vaccines. Louis Pasteur, done. ठीक है, अब नहीं भूल सकते all of his contributions. While you would have taken help of mnemonics and learned the 6 contributions, just remember the swan neck experiment in the wine and cheese industry and the car full of vaccines. Now, at the same time, in Britain, there was this guy with the sideburns. So the guy with the huge sideburns was a surgeon. Sideburns was a surgeon. And he is called as the father of modern surgery or antiseptic surgery. He is Joseph Lister. So you can remember antiseptic. What is the meaning of antiseptic? Sepsis. Sepsis means infection. Something which is antisepsis is anti-infection. Why do you put Dettol on a scraped knee? Because you can get infection. So you put an antiseptic, so sepsis can't happen there. Okay, it is anti-infection, anti-life. Antisepsis is when you prevent infection. So all of the surgeries which are conducted today, you are well aware, are antiseptic surgeries. Yeah, OT is completely clean. All the instruments you use are completely sterilized. The surgeon is wearing these masks and gowns. Nobody is allowed from outside. Even air is not allowed. On the doors of OTs, there are filters which completely filter out the air coming inside. So now the surgeries that we perform are completely sterile environments, and no amount of sepsis is tolerated. But at that time, if you look at this image, surgeries used to get conducted in open time, in open places. And at that time, the surgeons used to believe that since the patient's blood, mucus and everything will stick to my clothes will become dirty. So the surgeons used to wear dirty aprons and dirty clothes to perform these surgeries. And almost every patient used to die because of these infectious complications. Now, Joseph Lister was the first guy. He read the work of Louis Pasteur and he realized that there are chemicals, there are substances which can be used to make our surgical instruments also sterile. So he had a few compounds. Phenol which we use to wipe our floors even today, that is carbolic acid. So he made phenol. And you know, he gave which compound? Listerine. All the mouthwash that we use even till date, a lot of you must have used it today also. That Listerine has become a brand now. But he, Joseph Lister, gave us Listerine using phenol and carbolic acid. The phenol which we use, the phenol we use to clean our floors now, because now we have more advanced techniques, but at that time he was the first guy. So what did he do? He took this carbolic acid and he dipped his surgical instruments in it for some time. Then he took them out and he performed surgeries on 2 wounded soldiers. And the soldiers were fine. No infection happened on the soldiers. So he said that the surgeries we perform should be antiseptic. And he wrote it in a very, very famous journal, the Lancet. If somebody's paper gets published in Lancet even today, it's a big thing. And you know what the Lancet told him? Of course, they told him that you're ridiculous. No such thing happens. What are you talking about? If we dip our instruments in this chemical, what will happen? Germs are everywhere. But then now, of course, we understand his importance, and world over, of course, antiseptic surgeries are performed. So just a way to remember: Listerine, Lister, phenol, and the guy with the sideburns is a surgeon. So he is a father of modern surgery or antiseptic surgery. Let's go back to the rivalry. Let's go back to the rivalry. Who is he? Louis Pasteur, grumpy old man. Please remember the images, because sometimes they may give you images in your exams to be able to identify. Who is he? He is Robert Koch. He was a German guy. They used to hate each other. What was Robert Koch doing? Robert Koch was doing a very, very important work on TB. So the ways to remember is everything Koch's. What did Koch give? Koch gave Koch's phenomena. Okay. Koch gave Koch's postulates. Postulates means theory. It is my postulate that India is going to win against Pakistan in today's cricket match. I am postulating, okay? Theory. He gave us the Koch's bacillus or the Koch's bacteria. What is Koch's bacteria? It is the TB bacteria. Sometimes if you have people around you who have TB, if you have seen the notes of any doctor, they don't like to call it TB, a patient of TB. They say patient of pulmonary Koch's. Because Koch or Robert Koch is attributed with discovery of TB. So we call the disease also after him, we call the bacteria also after him. So whenever you say pulmonary Koch's, you are talking about TB. So Koch's bacillus or Koch's bacteria, TB he discovered, Koch's postulates he gave, Koch's phenomena he gave, and like I had said, while this guy was telling us all about the liquid broth, he poured some agar on it and he gave us the solid culture media, the agar plates which you might have seen even in the labs when you go. Robert Koch gave us that. So on the solid culture media, he was growing some organisms. Those organisms Louis Pasteur was using to make vaccines against. Okay, so one and one we shall look at Robert Koch. So if one scientist you want to remember from today's story, it should be Robert Koch, because they never get tired of asking this question on Koch's postulates. And once and for all we will read it today, and never ever again will we revise it, because it will get very boring otherwise, right? First, let's talk about Koch's phenomenon. What did he say? He took this bichara guinea pig. This guinea pig is sad looking, na? He has sad eyes. Why? Because he already has TB. He is already diseased. Which disease does he have? He has TB. What did Robert Koch do? Very, very cruel man. On top of the disease, he injected the TB bacillus inside him. Okay, already the guinea pig had TB. He then injected more TB inside him. What should have happened? He should have died, bichara. He realized that he produced the hypersensitivity reaction. Hypersensitivity reaction. So, if you know, there is a test called as the Montu's test, or the test for TB, in which they inject something in your arm and then it swells up and forms a wheal, and then after 2 days they take a measurement of this. That is the concept behind diagnosis of TB. If you already have the disease TB in your body, and if I take some protein of the TB bacteria, inject it into your arm, this particular area of your arm is going to hyperreact. is going to become hypersensitive because already the immune system is activated by TB. So the moment more TB comes in, it's going to get hyperactivated. This hypersensitivity of Koch's phenomenon is what we utilize for diagnosis of TB today. More than this, nobody will ask. Then what is Koch's postulates? One must know topic from this lecture. If you are sleeping, please wake up right now, sit up tight, because there are 5 important Koch's postulates that you have to remember, and it is highly irritating to revise them before the exam. So right now we will understand once and forever. Okay? 5 Koch's postulates of Robert Koch. Number 1, who is he? He's a mouse, okay? He took a mouse. He took a chuha in his lab and he took the solid culture plate which he had created. He said that this chuha has some lesions, he has some disease. This disease is because of some organism. Now the things which appear so obvious to us were not so obvious at that time. If I tell you कि आपको टाइफाइड के बैक्टीरिया से टीबी हो गया है, तो आप बोलोगे मैम पागल हो गई हैं. अगर मैं आपको बोलूं कि आपको टीबी के बैक्टीरिया की वजह से मलेरिया हो गया है, तो आप बोलोगे मैम पागल हो गई है. अगर मैं बोलूं कि आपको मलेरिया के पैरासाइट की वजह से टाइफाइड हो गया है, तो आप बोलोगे मैम पागल हो गई है, राइट? बट तब तक तो पता ही नहीं था ना कुछ भी. ही वॉज द फर्स्ट गाय हु टोल्ड कि अगर आपको टीबी की बीमारी है तो वो टीबी के बैक्टीरिया के आपके शरीर में घुसने से ही होगी. अगर आपके शरीर में टीबी का बैक्टीरिया है तो जो आपको बीमारी होगी वो ट्यूबरक्लोसिस ही होगी. ऐसा नहीं हो सकता कि मलेरिया का पैरासाइट आए और टीबी की बीमारी हो जाए, और ऐसा नहीं हो सकता कि टीबी की बीमारी हो और मलेरिया का पैरासाइट अंदर हो. Get it? Seems quite obvious, but that is the first Koch's postulate. Organism is constantly associated. Organism is constantly associated. क्या मतलब? जितने भी लोगों में टीबी की बीमारी होगी वो टीबी के बैक्टीरिया की वजह से ही होगी. विद द डिजीज. ऑर्गेनिज्म इज कॉन्स्टेंटली एसोसिएटेड विद द डिजीज. जब भी किसी भी चूहे में ये वाला लीजन आएगा, एक्स बैक्टीरिया की वजह से ही आएगा, वाई बैक्टीरिया की वजह से नहीं आ सकता. फर्स्ट कॉक्स पॉस्टुलेट. फिर अगर इस लीजन को उठा के मैं इस कल्चर प्लेट पे डालूं तो ये जो ऑर्गेनिज्म है इसको मैं प्योर फॉर्म में आइसोलेट कर सकती हूँ. Second Koch's postulate is organism can be grown or isolated, मतलब उसको निकाल सकते हैं in pure culture. So if I take a patient of TB and I take out the bacteria of TB, I should be able to grow it on the culture plate. Third Koch's postulate. If I take the bacteria from this culture plate, and what is this? This is a normal healthy mouse. नॉर्मल हेल्दी होस्ट है ये, है ना? अगर इस बैक्टीरिया को उठा के मैं इस माउस के अंदर डाल दूं, ठीक है, अगर इस कल्चर से उठा के मैं ऑर्गेनिज्म को एक हेल्दी होस्ट में डालूं तो होस्ट में जो लीजन प्रोड्यूस होंगी वो सेम होंगी एज दिस. कोई नई बीमारी इसको नहीं होगी, राइट? सो ऑर्गेनिज्म इनोक्यूलेटेड इन अ हेल्दी होस्ट प्रोड्यूसेस द सेम disease or same lesion is again produced in a different, this is chuha number A, this is chuha number B. Chuha number B got the same disease as chuha number A, okay? Same lesion will be produced. Koch's postulate number 3. If I again take the lesion from chuha number B, put it on the culture media, same organism in pure culture, I will get again. अब सुनने में ऐसा लग रहा है कि रॉबर्ट कॉच अंकल के पास कोई काम नहीं था, जैसे हम लोग अपने आंसर शीट भरते हैं ना, इज वॉज दिस दैट देम वर करके. रॉबर्ट कॉच वॉज द गाय हु वॉज फ्रॉम जर्मनी, हिज नेम वॉज कॉच, ही गेव द कॉक्स पॉस्टुलेट्स. कॉक्स पॉस्टुलेट्स आर द पॉस्टुलेट्स गिवन बाय रॉबर्ट कॉच हु वॉज द गाय फ्रॉम जर्मनी. हम लोग ऐसे ही अपनी आंसर शीट भरते हैं ना, ऐसे ही लग रहा है कि रॉबर्ट कॉच भी कुछ ऐसा ही गेम खेल रहे थे, सेम बात को बार बार बोल रहे थे. बट दिस इज द कॉक्स पॉस्टुलेट्स विच द वर्ल्ड डिड नॉट नो. And this is the basis of understanding all infectious agents even till today. इसी के बेसिस पे हमारी लेबोरेटरी डायग्नोसिस बनती है, इसी के बेसिस पे हमें इन्फेक्शन समझ में आते हैं, इसी के बेसिस पे हमें समझ आता है कि ट्रांसमिशन होता है इन्फेक्शंस का, राइट? सो कॉक्स पॉस्टुलेट नंबर 1, ऑर्गेनिज्म कॉन्स्टेंटली एसोसिएटेड विद द डिजीज. Number 2, organism can be grown in pure culture. Number 3, if I take the organism from the culture, inoculate inside a healthy host, same lesion is going to be produced in the healthy host. Postulate number 4, if I again take the lesion from the host, put onto a fresh new culture plate, same organism I am going to grow in the culture plate. Okay, what is the fifth Koch's postulate? Now all of us know that once we get a disease, antibodies are produced, but at that time we did not know. So he said that antibodies, postulate number 2, that antibodies are produced against the organism in a body of the infected host. These antibodies will not be found in the body of a healthy host. These antibodies will only be found in the body of the infected or the host with the lesion. 5 Koch's postulates, done and dusted for the rest of your life. Okay, rewind and listen to what I have said again, and please tell me at the end of the year that you do not have to go back to these postulates again. Done. Of course there are exceptions to life. ऐसे कैसे सारे organisms रॉबर्ट कॉच की बात को मान लेंगे? Of course exceptions तो होंगे ना, जिंदगी, biology is a science of exceptions. Of course. 2 प्री रिक्विजिट हैं कॉक्स पॉस्टुलेट्स को प्रूव करने के लिए. किसी भी बैक्टीरिया पे अगर आप कॉक्स पॉस्टुलेट प्रूव करना चाहते हो तो 2 रिक्वायरमेंट्स हैं. आपको समझ आ रही है? एक तो कोई चूहा या कोई एनिमल मॉडल होना चाहिए, कोई लैब एनिमल होना चाहिए जिसके अंदर वो बैक्टीरिया बीमारी करता हो. दूसरा, हमारे पास ऐसा कल्चर मीडिया होना चाहिए जिसमें वो बैक्टीरिया ग्रो करता हो, आर्टिफिशियल कल्चर मीडिया. ठीक है, तो या तो बैक्टीरिया के पास कोई कल्चर मीडिया ही नहीं है, हम उसे किसी आर्टिफिशियल कल्चर मीडिया में ग्रो ही नहीं कर सकते, तो उन बैक्टीरिया पे कॉक्स पॉस्टुलेट प्रूव नहीं कर पाएंगे. या फिर कोई एनिमल मॉडल ही नहीं है जिस पे हम प्रूव कर सकते हैं, किसी एनिमल में वो बीमारी नहीं करता. अब इंसानों में तो बीमारी करा के कॉक्स पॉस्टुलेट प्रूव नहीं करेंगे. So 3 important bacteria, and this becomes your MCQ. Of course, this becomes your MCQ. 3 important bacteria on which Koch's postulates could not be proven. अब ये जो इन्होंने प्रूव किया था ना, मैं तब से क्या बोल रही हूँ, टीबी, कॉक्स बैसिलस टीबी, सारा काम टीबी पे कर रहे हैं. तो कॉक्स पॉस्टुलेट्स भी इन्होंने टीबी पे ही प्रूव करे थे. टीबी बिलोंग्स टू माइकोबैक्टीरियम. अब माइकोबैक्टीरियम फैमिली का ही एक और कज़न है. एक हमेशा एक कज़न होता है ना फैमिली में, जो बताए रास्ते पे नहीं चलता, वो अपना कुछ अलग ही. नेवर टर्निंग बैक टू कॉक्स पॉस्टुलेट्स, डन एंड डस्टेड. सो गुड टू नो अर्पण, अमेजिंग टू हियर दिस. एक हमेशा फैमिली में एक ब्लैक शीप होता है ना, एक ऐसा कजन जो किसी उस रास्ते पे नहीं चलता. फैमिली में सब डॉक्टर हैं, वो गिटार उठा के म्यूजिशियन बनता है. ऐसा ही एक बैक्टीरिया है माइकोबैक्टीरियम लेप्री. अब आप सबको पता ही होगा, लेप्रेसी कराता है, लेप्रेसी जिसको कोड बोलते हैं हिंदी में, राइट. ये माइकोबैक्टीरियम टीबी जो बहुत प्यार से कॉक्स के सारे पॉस्टुलेट्स मान रहा था, उसी का कजन माइकोबैक्टीरियम लेप्री ने बोला, भाड़ में जाओ, मैं ग्रो ही नहीं करूंगा. आई विल नॉट ग्रो ऑन अ कल्चर मीडिया. So since we could not grow him on culture media, Koch's postulates नहीं लग पाए. Another bacteria, Treponema pallidum. Treponema pallidum. He causes syphilis. Syphilis is a sexually transmitted disease. महामारी हजारों लाखों साल से इंसानों को मार रही है, आज तक मार रही है, कुछ नहीं कर पा रहे हैं हम, right? Syphilis is a sexually transmitted disease, biggest killer of mankind, killing mankind even today. Both of these bacteria cannot be grown on artificial culture media. तो क्या बना? LT. यहाँ कौन आएगा? मैं तो सिर्फ इंसानों में बीमारी कराऊंगा, मुझे चूहे बहुत पसंद है. I will not cause disease in animal model. Neisseria species. क्या बोला? No. LTN. क्या बना एक्सेप्शंस का निमोनिक? LTN. लेप्री, ट्रिपोनीमा एंड निजीरिया. ओके, लेप्री, ट्रिपोनीमा, निजीरिया. अब कल्चर मीडिया कराया था ना, कॉक्स फिनोमिना देख लिया, कॉक्स पॉस्टुलेट देख लिया, कॉक्स बैसिलस देख लिया, और क्या किया रॉबर्ट कॉक्स ने? सी फॉर कल्चर. किसका कल्चर मीडिया? अब भाई चूहों से बहुत प्यार लग रहा है मुझे तो, है ना, हर जगह चूहे, तो जहां चूहे वहां बिल्ली भी. सो फ्रॉम माउस यू कैन रिमेंबर कैट. 3 बैक्टीरिया ही आइडेंटिफाइड. कैट. 1 is of course the TB bacteria, ना, of course it is TB bacteria. इतनी देर से I am screaming it's TB bacteria. What are these 2? Cholera. क्या बोला था मैंने, ये बेचारे उनको उगा रहे थे और वो कार में वैक्सीन बना के नाम कमा रहे थे। कॉलरा उन्होंने उगाया, इन्होंने वैक्सीन बनाई। एंथ्रैक्स इन्होंने वैक्सीन बनाई, तो ऑब्वियसली कैट का ए बनेगा एंथ्रैक्स। इनफैक्ट द फर्स्ट बैक्टीरिया जो उगाया गया था वो एंथ्रैक्स बैक्टीरिया था। सबसे पहले एंथ्रैक्स का बैक्टीरिया बैसिलस एंथ्रासिस उगाया गया था, उसके बाद टीबी। क्योंकि आगे हम पढ़ेंगे कि एंथ्रैक्स का बैक्टीरिया बहुत ही प्यारा बेचारा जायंट, आपने देखा होगा जो जायंट्स होते हैं वो बड़े बेचारे प्यारे होते हैं, ऐसी एंथ्रैक्स बैसिलस इज अ जायंट बैसिलस, बट ही इज वेरी वेरी बेचारा, कहीं भी ग्रो कर जाता है, इनक्यूबेटर नहीं चाहिए, कुछ नहीं, कहीं भी छोड़ के चले जाओ, आराम से ग्रो करेगा। जब हम पढ़ेंगे तब इससे मिलेंगे। अभी के लिए एमसीक्यू हो गया हमारा, सॉलिड कल्चर मीडिया में किसको ग्रो किया, कैट को, ठीक है, कॉलरा, एंथ्रैक्स एंड टीबी। रॉबर्ट कॉक फिनिश्ड। अब इतनी आसानी से तो आपको छोड़ेंगे नहीं, है ना। कौन लिख रहा था, अर्पण मैम, पढ़ लिया। अब एग्जामिनर क्या बोलेगा, एग्जामिनर बोलेगा अनुषा मैम से पढ़ के आए हो ना, ये बताओ मॉडिफाइड कॉक्स पोस्टुलेट्स बताओ। इनको हम मॉलिक्यूलर कॉक्स पोस्टुलेट्स भी कहते हैं। इससे पहले तुम्हें रोना आए मैं बता देती हूँ, इतनी मुश्किल बात नहीं है, ओके, डोंट वरी, डोंट क्राई, कर लेते हैं खत्म, समझ आ जाएगी बात। मॉडिफाइड एंड मॉलिक्यूलर कॉक्स पोस्टुलेट्स में हमने क्या किया, नाउ देयर वाज नो टर्निंग बैक, देयर वाज अ लॉट ऑफ रिसर्च हैपनिंग, और रिसर्च करते करते हम पहुंच गए कहाँ, हम पहुंच गए जीन्स के लेवल पे। अभी तक इन्होंने तो बोला कि भाई ऑर्गेनिज्म होना चाहिए, ऐसा डिजीज करेगा, ऑर्गेनिज्म ग्रो कर सकते हैं। अब लोगों ने बोला कि ऑर्गेनिज्म होने से बात नहीं बनेगी, पर्टिकुलर जीन्स होने चाहिए, विरुलेंट जीन्स। विरुलेंट वर्ड इज डिराइव्ड फ्रॉम विरिलिटी। विरिलिटी का मतलब होता है पावर, स्ट्रेंथ, स्टैमिना, विगर ऑफ लाइफ। तो अगर कोई बैक्टीरिया मरा पड़ा सा बेचारा है, वो डिजीज नहीं करेगा। कुछ बैक्टीरिया के पास एक स्ट्रेंथ और लाइफ, एक विरिलिटी होनी चाहिए, डेंजरस उसका नेचर होना चाहिए। उस डेंजरस नेचर को प्रोवाइड करने के लिए क्या होंगे, आपके कुछ जीन्स होंगे, ठीक है। दीज जीन्स, विरुलेंट जीन्स आर कॉन्स्टेंटली एसोसिएटेड विद पैथोजेनिक बैक्टीरिया। अब कुछ बैक्टीरिया होते हैं गुड बैक्टीरिया, कोई कमेंट बॉक्स में थोड़ी देर पहले लिख रहा था, हाउ कैन वी जस्टिफाई दैट बैक्टीरिया आर गुड फॉर अस। सम बैक्टीरिया विच आर गुड फॉर अस आर नॉट, इज देयर समथिंग लाइक एनाटॉमिकल नॉमेनक्लेचर हियर, आई डू नॉट अंडरस्टैंड योर टर्म बेटा, ठीक है। तो अब नाउ दिस इज एसोसिएटेड विद पैथोजेनिक बैक्टीरिया, द बैक्टीरिया विच कॉज डिजीजेस, only they will have these virulent genes and the other good bacteria will not have. एक बैक्टीरिया होता है क्लोस्ट्रीडियम डिफिसिल, अभी नाम से कंफ्यूज नहीं होंगे। एक तरह का बैक्टीरिया है हमारी गट में जो कि फ्रेंडली होता है, हम सबकी गट में नॉर्मली प्रेजेंट होता है। कभी कभी किसी किसी बैक्टीरिया का दिमाग खराब होता है और वो कुछ टॉक्सिन के जीन बनाने लगता है, और जब वो उन टॉक्सिन्स के जीन बनाने लगता है तो टॉक्सिन्स को निकालता है और हमें डायरिया करा देता है। नाउ वी हैव नॉट ईटन दैट बैक्टीरिया फ्रॉम फूड आउटसाइड, बैक्टीरिया वाज ऑलरेडी इन आवर गट बट ही वाज नॉन पैथोजेनिक, लिविंग नाइसली। सम डे सम gene got activated and he started producing toxins. Now we are in a soup, we have gotten diarrhea. So they said these virulent genes are associated with the pathogenic bacteria constantly and not associated with normal bacteria, okay. Now virulent genes, if there is some mutation and there is loss of these virulent genes, अगर मैं कुछ म्यूटेशन कराऊं, कोई रेडिएशन डालूं और ये जीन म्यूटेट होके लूज हो जाए, सो जो पैथोजेनिसिटी है इस बैक्टीरिया की, मीन्स द एबिलिटी ऑफ द बैक्टीरिया टू कॉज द डिजीज इज गोइंग टू रिड्यूस। Hence it proves that it was this gene which was responsible for causing the disease. First modified Koch postulate says virulent genes are constantly associated with pathogenic bacteria. Second postulate says if there is a mutation and these genes are lost, pathogenicity of the bacteria reduces. Third postulate says if I reverse this mutation, if I reverse this mutation and the gene is restored, the pathogenicity comes back. The bacteria becomes dangerous again. Pathogenicity is restored. So this is a reversible damage. This is not irreversible damage. एक तरह से अगर starfish का मैं हाथ काट दूं तो कुछ टाइम के लिए वो हैंडीकैप हो जाएगी, लेकिन कुछ दिन बाद वो उसे दोबारा ग्रो कर लेगी और फिर से चलने लगेगी, right? That is what is with the genes. Genes are present in pathogenic bacteria. Mutation happens, pathogenicity decreases. If at all the mutation is reversed and the genes come back to life just like the wild type bacteria, then the pathogenicity is restored. Then three postulates में modified Koch postulates would have gotten over, but then how would they have made your life miserable? They had to make your life miserable. They went one step further. They said merely presence of gene is not enough. The virulent gene should get expressed during the course of illness. I am a person. Suppose I have the genes of an angry person inside me. Those genes are present, but they are lying dormant. They are not getting activated. I seem like a non-pathogenic teacher to you. I am not a dangerous teacher. Someday, those genes start expressing themselves and I become red eyes, horns come out, frothing from the mouth. I become a complete monster. Then I become pathogenic. So these genes will only be present in pathogenic bacteria. And whenever, due to some stimulus, the genes get activated or expressed, it is only then that the disease will happen. That is Koch's postulate number 4. Number 5 is more or less derived from antibodies. While Robert Koch told that antibodies are produced, they said specific neutralizing antibodies are produced. Means now we have gone at the molecular level. Na? At the molecular level we have gone and we have also understood the property and the shape of the antibodies, everything we are going to read in microbiology. So now we know that there is one particular site on the antibody to which one particular protein of one particular bacteria binds. If my body produces antibodies for malaria, the same antibodies will not work against tuberculosis. Okay, these are specific antibodies. Fifth modified Koch's postulate is specific antibodies. So for molecular Koch's postulates, we are talking about genetic level. Okay, so for Koch's, remember this image. If you just take a mental picture of this image, then you will never forget Koch's postulate. Chuha, culture plate, chuha, culture plate, antibody. Now you can make up the Koch's postulate on your own. Then over here, gene, lost, regained, expressed, specific antibodies. Done. Mind map, finished. Koch's postulates, done and dusted. Modified Koch's postulates, done and dusted again. Arpan, I hope you're watching and I hope modified is also done and dusted. If it is done and dusted, please write. ठीक है, 10 मिनट और चलेगा. एक हमारा और रह गया है पर्सन. दैट्स इट. अब ये कौन है? रॉबर्ट कॉच. कैसे याद रखेंगे? लुई पैस्चर की शक्ल कैसे याद रखी थी हमने? ग्रम्पी ओल्ड एंग्री मैन. दूसरा क्या? इतनी नफरत, बाल ही उड़ गए. इतनी नजर लगाई इन्होंने कि इनको चश्मा भी लग गया और इनके बाल भी उड़ गए. सो मच हेट्रेड ही वाज गिविंग हिम, राइट? इतनी नजर लगी, बाल उड़ गए. अब लेकिन इन्हीं के जैसे दिखने वाले एक और साइंटिस्ट थे, वो थोड़े यंग थे, उनको नजर कम लगी थी. कौन थे वो? पॉल एरलिक. देखोगे ध्यान से, बोथ ऑफ देम लुक क्वाइट सेम, राइट? सेम तरह का चश्मा, सेम तरह की बियर्ड. क्या फर्क है? इनके पास बाल नहीं है, इनके पास बाल है, क्योंकि इनको मिल रहा था बहुत सारा प्यार. बिकॉज Robert Koch and Paul Ehrlich were BFFs. They were best friends forever. They were colleagues and they were working together. Of course, किस पे काम किया? उन्होंने टीबी पे काम किया. टीबी से ही ये ऑब्सेस थे, टीबी से ही ये भी ऑब्सेस हो गए. ये है वो टेस्ट, मॉन्टूस टेस्ट, जो मैंने आपको बताया था हम इंजेक्ट करते हैं. दे डिस्कवर्ड दिस टेस्ट ऑल्सो. कॉक्स फिनोमिना के बेसिस से इनको लगा शायद हम ट्रीट कर देंगे पेशेंट्स को, पर बाद में पता चला इससे डायग्नोसिस बना सकते हैं. अब पॉल एलरिच, एक और फोटो आपको दी जाती है. कैसे कंफ्यूज नहीं होना है, इनके पास बाल हैं. लुक्स एग्जैक्टली लाइक रॉबर्ट कॉच, हैज हेयर. ओके, इनको बोलते हैं, पॉल एरलिच को बोलते हैं फादर ऑफ कीमोथेरेपी. फादर ऑफ कीमोथेरेपी. कीमोथेरेपी मतलब therapy matlab treatment, chemo means chemicals. Today we treat the diseases by giving certain chemicals which are the antibiotics or which are the drugs, right? These drugs, these antibiotics of what? These are chemicals. Paracetamol is a chemical. So we treat the antibiotics using these chemicals, that is called as chemotherapy. He was the first guy who ever said that you can give the chemicals to treat the disease. Now this is now while with Louis Pasteur and everyone was distracted by the organisms and all organisms. He came in and he said, wait a minute, use chemicals to kill these organisms, right? So thank you, Dala. I'm happy you like the explanation. So Paul Ehrlich is the father of chemotherapy, and I had talked about syphilis, right? The biggest killer of mankind is a sexually transmitted disease. He used a compound which is an arsenic compound called as sulvarsan. This sulvarsan he gave in patients of syphilis to treat, and he called this the magic bullet. Today we use this word magic bullet for antibiotics because these antibiotics are these small little bullets which are so powerful. You all must have heard the name of Alexander Fleming. What did he give? Penicillin. This penicillin was the magic bullet which saved the lives of thousands of soldiers during World War II. While everyone would die in world wars, this single penicillin saved many soldiers. So these were magic bullets. So he was the first guy who told it. Then of course, what is this? This is a antibody. This antibody has a side chain. So Ehrlich gave the side chain theory of antibodies. He also gave the side chain theory of antibodies. Name is Ehrlich. He gave the Ehrlich phenomenon. Now what is, why Robert Koch gave Koch phenomenon. How will the best friend stay behind? He will also tell something. What will he tell? He was focused on antibodies. He said, he told about the toxin-antitoxin reaction, meaning that if there is some toxin which is produced, you can give some antitoxin to neutralize the effect. There is a bacteria called as diphtheria, causes a deadly infection of the throat. It produces these toxins. Whenever such a patient comes to me, I give injections of antitoxin and the patient is fine. So he was the father of chemotherapy, and he said not just these chemicals, you can also use antitoxins against toxins to kill the effect of toxin. That is Ehrlich's phenomenon. And of course, Ehrlich, he gave Ehrlichia species. If best friend was discovering so many microorganisms called as cat, he also had to discover a microorganism, Ehrlichia species, right? Very interactive, easy to understand. Okay, thank you. Right? So this is Ehrlichia species. Then he also gave the acid fast staining, which is called as the Zn staining. आप लोग अपनी class में जाओगे एक बार, so you will see. So the stain which we use to detect the TB bacteria is called as acid fast stain. So both of the best friends were working on TB only, so he gave acid fast staining. So now you will never forget. Best friend with Robert Koch obsessed with TB, he gave the stain. He said put this dye on the bacteria and you will be able to see it. So acid fast staining also he gave. So Ehrlich, father of chemotherapy, side chain, Ehrlich phenomena, toxin-antitoxin, Ehrlichia species of bacteria, and acid fast staining. Finished. Who is he? Painting. A.V. Leeuwenhoek, father of microbiology, little animalcules. Revising. Who is he? Sideburns. Sideburns surgeon. Joseph Lister, Listerine phenol. Sepsis, father of antiseptic surgery, surgeon. Okay, who is he? Grumpy old man. Louis Pasteur, pasteurization, fermentation, heat sterilization, germ theory, liquid media, vaccine. Which vaccine? Car full of vaccine. Cholera, anthrax, rabies. Done. Who is he? I hate him. नजर लगा के बाल उड़ा दिए. Robert Koch. Koch phenomena. Koch postulates. Koch bacillus, TB bacteria. Solid culture media. Culture of cat. Cat. C is cholera, anthrax and TB. BFF. Paul Ehrlich, father of chemotherapy. Side chain theory. Side chain theory. Ehrlich phenomena, antitoxin, toxin, Ehrlichia species, and as TB stain for TB, which is acid fast stain. Finished. All of the important scientists finished. As promised, end of class, we will have the food for thought. If Paul Ehrlich is credited with the discovery of acid fast staining, then why is the stain called as Ziehl-Neelsen stain? Ziehl-Neelsen stain क्यों बोलते हैं? A hint I will give you. See, Paul Ehrlich is everything CH, na? So CH is also cheating. Cheating. इनके साथ cheating हुई थी. He got cheated and took away the name for acid fast staining. Why? Because Zeal and Nelson did something which made them take the credit for acid fast staining. So this is your food for thought today, that if he was the one who discovered, then why do we call the stain as Zeal-Nelson stain? What did Zeal and Nelson do? Please find out. Of course, the second food for thought is in fact a INICT previous year question. Okay, it was asked in INICT. Of course, we will go back to microscopy, first part of the lecture. Right now, it feels like it was done yesterday, but koi baat nahi. Which microscopy technique provides the best contrast? The best contrast. Is it dark ground microscopy? Is it fluorescent microscope? Is it phase contrast microscope? Or is it electron microscope? Which provides the best contrast? Okay. If you want to give the answers, you can give them to me right on my social media. Okay, I always give you that you can ping me on my social media and share the answers with me. Otherwise, of course, we are going to discuss the answers in the next lecture. And like I always say, stay in the game. That's all that you need to do to pass, to survive, to thrive, to win. You simply need to stay in the game. It was amazing to have this session with you guys. I'm so glad that we could finish it relatively on time. These were two intense, huge topics that I had taken up to be covered in a single class. With this lecture, we have shubharambhed and gone straight into our core microbiology. We had started discussing general microbiology. So it was amazing interacting with you guys. Thank you so much for all the lovely comments that you're leaving. So that's it. I will see you guys next week. And it was amazing. Thank you so much. Bye-bye. Any questions, please reach out to me on my social media. Thank you. Thank you.